wyłączone. Dobra, wyłączone. Right?
Yes, you can continue, but you have to switch on again your presentation.
Sure. So we have covered this part and this part. These are some of the partial aims on these 4 locations: Lokecka spoil heap, Bohemia 1, Antonin, and Litov spoil heap. They were all established during different times, and they vary in vegetation and time of establishment, but they all come from the coal mining region of this area. The methods for the research vary from like paper to paper and analysis to analysis, so I did not include them in this presentation. But to begin with, my former supervisor had an idea that in order for me to get familiar with the topic, I need to do some review papers and gather some information on it. So these are just 2 publications that we managed to put together. And from those, me, who wasn't initially in this part of the topic, managed to get some general ideas of the problems that post-mining sites and coal post-mining sites in particular are facing. So there are some big physical, chemical, and biological issues that may or may not be familiar with. The biggest issue on post-mining sites is how to successfully establish vegetation, obviously, if you're trying to revegetate them. And through this, I gained some information on some common tree species that are used for the process of reclamation, restoration, remediation, revegetation. There's very big confusion about the terms there. It's also explained. And there's nowadays an ongoing big debate about passive versus active revegetation practices. Active revegetation practices are mostly considered to be technical reclamation, which is also included in legislation of many countries. And nowadays they're debating whether that's good or bad, because some research have proven that passive revegetation, which means revegetation by natural succession processes, is sometimes much cheaper and much better for mainly biodiversity, but some other things in the long run in general. And regarding soil formation on post-mining sites, there was a debate about the speed. It really depends on the parent material of your post-mining sites in general, but sometimes the general idea is that soil formation on post-mining sites can sometimes be much faster than in nature, because the pedogenetic parent material is already turbated and started weathering in some way, so the soil is a little bit more prepared to start the process of soil forming there. There is also a debate when you look at the literature about the so-called biological versus pedological soil development. So biological soil development is mostly based on observing the organic matter and nutrient changes and cycles, whereas pedological soil development is generally found to be you dig a hole in the ground and try to see which horizons are there. So through the formation of diagnostic soil horizons. However, that usually takes ages to have them. So most of it is the biological soil development observations. And there's also been some current trends in post-mining research, like countries which are leading the research, et cetera, mainly focusing on carbon sequestration and nutrient flows. Now, for Sokolov region, we sort of had an idea and a preliminary hypothesis that our soil will tend to evolve from pure technogenic post-mining technosoil to maybe a forest cambisol under different forests and trees. So, some of the main parts of my research you can find in these 2 publications. Number 3 marked here focuses on just the soil aspect of soil formed under different trees of the same establishment age some 50 years ago in Sokolov region at Antonin Forest Arboretum. Spoil heap. Both of these researches were done there. Just the number 4 was done in cooperation with the guys from the Faculty of Forestry of Czech University of Life Sciences. So they assessed everything like production potentials, biodiversity, and soil properties, which was my part and contribution to this research. So there are 2 of them. This one focuses mostly on soil. This one focuses on other aspects as well. And we found certain things there. So this is the Antonin Forest Arboretum there, established in the early 70s after the mining. And it's a really beautiful place to see. My research there was split. We tried to, this arboretum had over 220 different species and cultivars of different trees established there. So we managed to find 23 monocultures, like homogeneous stands that I can test the soils under. The broadleafs are marked light green, the conifers are marked dark green in this place. So these were some of the species that we've tested. All the broadleaf species were native to the area. From coniferous species, we had native larch, Norway spruce, and Scots pine. Everything else were introduced species there. And we've done some basic physical and chemical soil analysis on the soils under these trees. This is just so I wouldn't show you graphs, etc., which you can read in the paper. This is just some of the preliminary thin section microscopy showing you the porosity after 50 years of soil formed under European beech and under Norway spruce. And as you can see, the compaction levels under Norway spruce are much greater. You probably know why, because you are all somehow related to the forestry segment. Right, okay, so some of the main findings of the research, when you compared all the soil properties that were tested there, were that, as expected, some species like alder, beech, linden, pear, blue spruce, which is a little bit weird, and ponderosa pine have shown some good physical and chemical soil properties that are generally considered to be good. On the other hand, birch, larch, Norway spruce, mountain, Weymouth, and Scots pine did not do so well. And this graph shows, in a way, a split and the depth of the formed organomineral A horizon after these 50 years in the section. And you can almost clearly see the distinction between the conifers and the broadleafs. Like the conifers are on the left, under 3 centimeters of A horizon depth, and the broadleafs are mostly on the right with more than 3 centimeters of depth. Some exceptions here were blue spruce and black pine, and some exceptions from the broadleafs were hornbeam and birch. So this is just a principal component analysis of how they cluster and how they look, and there's also like a clear grouping trend between the broadleafs and conifers here. However, we did have 2 papers mentioned there. So what I showed here is just from soil properties perspective, from production potential and the actual health of the trees growing on at Antonin Forest Arboretum there. We have something like a conundrum. For example, Pitea pungens did really well from soil point of view. On the other hand, Pinus sylvestris did not do as well. From production potential and health of the stands there, Pinus sylvestris is one of the absolutely best growing tree species of the Arboretum, together with larch and some others. There's a potential debate that it also sort of depleted the soil properties and embodied it into their actual body structure. So that's one of the things, but it also shows why you need the sort of comprehensive approach, and you cannot just observe the soil without doing other things. On the other hand, Pizea pungens has a very reduced stand. It's dying off from climate and pathogen threats there, and there's just a few trees left there. So it's a thing worth taking home. In other parts, we try to assess the effect of time on soil forming processes. This paper has already been published. This one is under second round of review. So within this region, we took 3 stands of 3 localities of different age, these post-mining sites. So the youngest one was monoculture of alders planted after some 30 years ago. The other one was a 50 years clear maple stand, and the third one is a very old mining site and one of the oldest in this region. It's around 90 years old, and it's a combination of cherry and maple. So what we did is we dug some deep soil profiles there and trying to assess the horizon boundaries formation, and took some samples of soil from these pits to analyze them in like their chemical and physical properties. The parent material of the entire region, apart from one spoil heap, is consisted of cypress clays, which are tertiary and quaternary clays, which you can clearly see this sort of leaf formation in them. And all of the profiles tested basically exhibited similar horizon formation. Organomineral, cambic, and then the parent material. They're basically proving our initial theory that it would shift from a technosol to a cambisol there. So this is just a picture showing 2 soil profiles from the oldest mining sites and their profile development. We dug in 3 locations 6 profiles in total. One was always done on plain terrain, one was done on a slope, because we assumed that on plain terrain the accumulation of organic matter and everything else would lead to actually slightly thicker horizon formations. So in the end we got this, so 30 years, 50 years, 90 years, and you can see that with time all of these horizon differences are sort of what we expected. The one on plain terrain here was thicker in terms of all horizons here. This one was sort of inconclusive, and this one was inversed. However, Loketska on level terrain turned out to have a slightly different parent material in the chemical analysis tested, so that may have led to slightly slower development. Basically, with time, we had clearer horizon boundaries, more observable color differences, etc., and wider ranges of physical and chemical properties results there. What we got from this is what's pretty much known. The effect of soil organic matter accumulation and mineralization did improve soil quality. We also tried to test the quality of humic substances, and the oldest one did prove to be better than the others. The effect of slope, it sort of confirms our initial expectations, apart, as I said, from this part here. And other part of this research that my other colleague is doing her PhD thesis on is Litov spoil heap over there. And Litov spoil heap is very interesting in terms of most of the parent material is the cypress clays, which generally have pH of 8 and above when you test them. However, this particular spoil heap contains some shales, which contain pyrite, and pyrite has pyrite oxidation, so there are sulfuric acid flows, color changes, and dying off of vegetation. The lowest measured pH value there was 1.8, so see how hard it is. These are just some pine trees that unfortunately did not survive, so establishing vegetation there is pretty hard. However, we did a research. Some of our colleagues from our department did monitoring in like 100 points there in the region, and they did measurement of certain soil chemical properties in 1998. So we repeated this experiment in 2018, and we got some values from those. Generally, it should be published soon, we hope, but generally an increase of pH, organic carbon content, and slight improvement in quality of humic substances was noticed in 2018. So after 20 years, the conditions got better in most of the region, but some parts of the region got more severe acidification over time, leading to some potential aluminum toxicity, etc., etc. From a forestry standpoint, afforestation, mainly by deciduous trees, supported the improvement of these soil properties as was expected. But the issue is it has high pyrite content. The species there were chosen wisely when they initially did it. However, it's just there's so much pyrite oxidation and sulfuric acid flows that it is a bit problematic in the end. So conclusions out of combined approach to all of this is that the effect of time is clearly visible, and accumulation of soil organic matter has a very important role, obviously. From the standpoint of soil formation and soil quality, some long-lasting broadleaf species and black alder, which is a commonly used species for post-mining sites, are recommended. However, some suitable native coniferous species like Scots pine and larch did grow well there. Spruce was a little bit more problematic. And some suitable introduced species were black pine, Serbian spruce, and Douglas fir, and all three of these grow in the Antonine Forest Arboretum extraordinarily well. The effect of relief on soil formation was noticed and addressed, but a little bit of further research is recommended. And we still have some future publication plans and some results which have not yet been processed. Now, I originally do come from Serbia, and we had Serbian spruce here. Serbian spruce is an endemic relic in Serbia and is slowly dying off and moving to some very small parts of certain mountains. And when I first came to Czech Republic and saw that they did a plantation and saw the closed canopy of Pizea morica here, I was really puzzled and I really liked it. So it also grows well, and some other people, including my roommate here at the conference, were doing some research about Serbian spruce on this particular spoil heap, and the results are astonishing. So moving Serbian spruce from its native area a little bit to the north might be a way to preserve the species, and it works well sometimes in these conditions. Now, there are some other potential factors that we don't have time to assess, but here, the last slide is just a photo from a textbook we used back in the Faculty of Forestry, where an old professor of mine took a photo in the Sokolov mining region in Czechoslovakia at the time, and you can see his height compared to the established spruce trees. As we said, it started in like 72, 74, so this is how old they were in 81, and this is just a photo I took in the Serbian spruce forests in 2019, and this is the height of a man there, and you cannot see the tops there. So thank you very much for your attention, and if you have any questions, feel free to ambush me and ask me.
Thank you very much. All the questions will be given to you at the end of the session when we'll have discussion now.
So hello everybody. Thank you for the introduction, and I am very happy to be here. It is a very beautiful venue, and everything is so well organized, so my compliments to the organizers. So, my name is Kristiina, and I'm from Estonian University of Life Sciences, and I have another presentation about bryophytes for you. Marcin already yesterday introduced his very interesting results, so mine are a bit different specialty. I am mostly concentrated on research on wind disturbance and the subsequent vegetation development. And today I'm talking about the response of bryophytes to wind disturbance and salvage logging approximately 20 years after the wind disturbance took place. So why are the bryophytes important? First of all, the bryophytes are able to colonize inorganic surfaces as pioneer species. Thus, they promote soil formation. They also fulfill numerous important functions in forests and participate in many key processes like water retention, fixation of nitrogen, carbon storage, nutrient immobilization, vascular plant facilitation or suppression, and so on. And bryophytes are often a bit overlooked in forest ecology, so I'm trying to fill this little gap. So we studied abundance, richness, diversity, and community composition of bryophyte species in wind-damaged plots situated in East Estonia. So here is Tudu plots, and here is Halliku. And we monitored the vegetation in moderately damaged plots. That means where nearly approximately half of the trees were downed during the wind disturbance. Heavily damaged stands where approximately all of the trees were downed, and heavily damaged and subsequently salvage logged stands. And we also included the relatively old forest stands as a reference group. So we expected to find that environmental characteristics such as microsite diversity and richness of tree species differ among treatments, and these differences are reflected in vegetation characteristics. So we also expected that to find that bryophyte diversity is greatest in moderately damaged stands because those sites were expected to have greatest heterogeneity in terms of different substrates available. We expected to find that control plots have intermediate diversity values, and we expected to find that the magnitude of change in bryophyte composition is correlated to the severity of disturbance. As we know that vascular plants tend to respond strongly to light levels and soil richness, bryophytes have a bit of different life strategies and traits, and they are known to respond strongly to heterogeneity of the environment. And that was our main hypothesis. So we made 100 square meters study plots, and the cover of each species was evaluated using the Brown Plunkett 5-class cover abundance scale. And we associated each species with a growth substrate using 5 different categories: intact ground, dead wood, live tree, pit and mound. And I think Marcin yesterday described hummocks and hollows. Well, it's the potato-potato situation. It's the same thing, just we name it mound and pit. And all of these different substrates have very different environmental conditions, and thus they can host different species. We also estimated visually the proportion of each growth substrate within the study plots, and we used 3 diversity metrics: species richness, Shannon diversity index, and Belows evenness index. One way ANOVA and Tüki harnessed significant difference tests were used in order to detect differences between treatments, and linear regression was used to determine the effects of these explanatory variables to response variables. And the permutational multivariate analysis and non-metric multidimensional scaling analysis were used to detect the compositional differences. So what did we find out? First of all, as we expected, substrate diversity index, which was calculated analogously as the diversity index for species richness is usually calculated, and this index was significantly higher in wind disturbed plots compared to control and harvested plots. And the multiple linear regressions revealed that all of the 3 metrics of biodiversity were highly correlated with differences in substrate diversity. And treatment was also an important factor. The richness of liverworts was best explained by the sole explanatory variable of substrate diversity, while masses additionally responded to treatment and the proportion of deadwood substrate. So here you can see the box plots. Altogether, we found 108 bryophyte taxa, including 81 mosses and 27 liverworts. We could identify 105 of them to the species level. And on average, each plot contained 42 taxa, and the species richness was highest in heavily disturbed plots and lowest in control plots. So, as you can see, here are species richness was significantly different between moderately damaged plots and harvested plots, moderately damaged plots and control plots, heavily damaged plots and harvested plots, and heavily damaged plots and control plots. So the overall species richness was highest in moderately damaged plots, which contained 85 species, and moderately damaged plots also had the highest number of unique species. So it means that there were 12 species that were not found in any other treatments. Heavily damaged plots contained 81 species with 9 unique species. Harvested plots contained 60 species, and control plots had similar number of species. So we also wanted to find out if some conservationally important species respond differently to the treatments. And we found that moderately damaged plots hosted the greatest number of species with high conservation value. There were 10 species altogether, including 2 species that were exclusively found in those plots. Heavily damaged plots hosted 7 species with high conservation value, and control plots and harvested plots had 6 and 3 such species, respectively. So most of the taxa were associated with the deadwood, which is not an unexpected result because I think everybody knows that bryophytes like deadwood a lot. 65 species were growing on intact ground, and intact ground also had the highest number of unique taxa. 58 species were found on mounds, 50 in pits, and 56 on live trees. And from the live trees, most species were found on spruce. There were, I think, 44 species. Betula pendula and Betula pubescens together hosted 36 species. Alder hosted a similar number of species, and so on. So here are the ordination plots. And the non-metric multidimensional scaling analysis revealed that diversity of substrates, treatment, and proportion of deadwood significantly influenced the species composition. So here is the deadwood, and here is the axis that shows the heterogeneity of substrates. So with the green asterisk, here are mosses, and with a red asterisk, liverwoods. And as you can see, harvested plots, well, there is some overlapping between these wind-disturbed plots and control plots. Harvested plots are quite different from these old intact forests in terms of species composition. And also it is quite interesting that these are the heavily damaged plots, and as you can see, they are quite similar to each other, but the moderately damaged plots are very different in terms of species composition, which means that they have a very high beta diversity. So in conclusions, nearly 2 decades of the storm disturbance, wind disturbed the stand, sustained bryophyte communities with great species richness, thanks to the high substrate diversity, habitat provision through an increase in quantities of structural elements such as deadwood is especially important for liverworts. And thus, if the aim is to promote bryophyte diversity, natural recovery should be preferred over salvage logging. But if the salvage logging is implemented, the possibility of leaving part of the biological legacies of the disturbance in situ should be considered. So, if we are interested in the full text, then this article quite recently got published. And if you do not have access to it, please email me or contact me via ResearchGate. I will happily share my research with you. And I have also done a similar study with vascular understory plants, so don't hesitate to contact me if you're interested. So thank you very much.
Thank you very much, Christina, from your brave hats from Estonia. The next topic about forest road networks. Surface deformation monitoring in forest road networks, a high precision UAV real-time kinematic approach. But Evangelia cannot make an oral presentation today in conference hall. But she has prepared the film about your study, so let's watch the film of Young Science.
And the title of my research project is Surface Deformation Monitoring in Forest Road Networks, a high-precision UAV real-time kinematic approach. Forest roads play a vital role in facilitating access to management and exploitation of forested areas, aiding in the efficient and cost-effective transportation of timber from the logging site to areas of concentration and processing, and supporting the development of mountainous regions. Therefore, the proper construction and maintenance of forest roads are imperative for ensuring smooth vehicular movement. The degradation of forest road network, particularly the erosion of soil, is a complex phenomenon with multiple components. Skid trails locally increase soil compaction, and forestry activities may cause sedimentation of surface water if best management practices are not implemented during forest operations. Harvesting method, number of machine passes, soil till, type, texture, and water content, among others, affect soil compression and rutting during mechanized logging, as forest operations can have a significant impact on the physical and chemical qualities of soil. The aim of the study was to develop a novel approach that identifies classes of surface deformation on a forest road using a fixed-wing UAV equipped with RTK technology compared to previous research where polycopters have been used. By utilizing this specific equipment, we aspire that the proposed methodology is characterized by faster implementation and high accuracy data collection for large-scale projects. The collected data can be utilized to improve the terrain's condition, streamline its administration, and facilitate the implementation of technical measures and interventions in specific segments. And let's proceed into materials and methods section and the study area. As seen in Figure 1, the research area is located in northern Greece in Chalkidiki Peninsula. We investigated the forest road for pavement deformations with a total length of 3,200 meters. The elevation is ranging from 154 meters to 233 meters, and the road traverses established agroforestry systems. To conduct the research, the following equipment and software were used: the SenseFly EBX, a fixed-wing unmanned aerial vehicle with an absolute accuracy of 1.5 cm, the SODA 3D drone photogrammetry camera, the eMotion flight planning software version 3, Pix4D Mapper, a photogrammetry program for professional drone mapping, ArcGIS desktop, an integrated suite of geographic information system software, and TR7 GNSS RTK receiver from SATLab in order to map the forest road. Next, we will analyze the methodology. Generation of Forest Road Surface Deformation Index. The primary goal of this study was to classify deformations occurring along the surface of the unpaved forest road. Our objective was to improve the condition of the road by facilitating more accurate management and providing measurement data for the implementation of technical structures and interventions in specific segments of the road. In order to finalize the generation of deformation classes for the road surface, the following elements were implemented: an orthomosaic map georeferenced to the Greek reference system EXA 87 provided a geospatially accurate representation of the study area, a DM that was employed to capture the bare earth surface, a DSM with a spatial resolution of 4 cm per pixel that served as presentation of both terrain and above ground features, and the polyline shape file of the road that included its spatial attributes. These datasets were imported into Esri's ArcMap software to proceed to further analysis and processing. The data collected by the EBX flight were imported into the Pix4D Mapper photogrammetric software. The specific software was used to generate the DSM and DEM. The generated products were then imported into the ArcGIS software, and using the focal statistics tools, we defined 3 box ranges in pixel sizes, 3 cm, 4 cm, and 5 cm respectively. The range option was used, which is not a physical unit, but rather a continuum of values that determines the level of smoothness observed on the forest road pavement. The next phase involved the reclassification of the box ranges for each pixel size into 20 distinct classes. The most effective range for detecting variations based on the topography of the road and its surroundings was box range of 4 cm. The initial group of 10 classes was created with each class having a width of 1 cm, and this was succeeded by an additional 9 classes, each with a width of 5 cm per class. The 20th class had values exceeding 55 cm, which were assigned to various objects such as trees, buildings, pillars, and steep slopes. These values were only taken into account for certain maintenance-related tasks. Following the classification of the elevational groups corresponding to the distortion seen on the route, further steps involved the calculation of the surface area associated with each class, as well as the calculation of the amount of material to be excavated and deposited for the road. Generation of a flood risk map. To create the flood hazard model for the surveyed area, the DEM acquired through surveying with the EBX UAV drone, together with a shapefile delineating the borders of the area, were imported into ArcMap. The structural method was employed for the assignment of stream-ordered branches within the hydrographic network. The processing steps included filling soil depressions, determining flow direction, estimating watershed flow accumulation, delineating the hydrographic network, segmenting branches, and assigning stream order numbers. Additionally, the identification and mapping of potential flood-prone areas was also included in the data processing workflow. This classification process is informed by various data inputs, including historical precipitation data, soil composition data, slope data of the area, and data on land uses and forms of cover. And we go to the results data and UAV data accuracy statistics. Geolocation error is the difference between the original and calculated image positions in the photogrammetry software. Table 1 assesses the quality of the image geolocation by presenting information on the percentage of geolocated and calibrated images that exhibited inaccuracies in the X, Y, and Z coordinates within predefined error ranges. Sigma indicated the standard deviation of the error in each direction was 0.022 meters, 0.019 meter, and 0.020 meters respectively. Furthermore, the root mean square error was found to be 0.022 meters, 0.019 meter, and 0.020 respectively. We proceed to the map generation. Following the processing and analysis of the data in the Pix4D Mapper software, the orthomosaic and the corresponding sparse DSM before any densification process were developed and are depicted in Figure 2. Key aspects of the study area are presented in Figure 3. These include the digital elevation model, the digital surface model, the slope map, and the aspect map. The collected data facilitated the generation of a comprehensive flood risk map seen in Figure 4. It can serve as a valuable visualization tool when considering the implementation of infrastructure projects within various sections of the forest road networks, providing an understanding of the specific flood risks associated with these areas. Based on the data analysis, it can be observed that the area mostly receives water from streams classified as 4th, 3rd, and 2nd, with a limited presence of 1st-class streams. The deformation seen on the surface of the road can be attributed to the shape of the hydrographic network and its distribution throughout the study area. In relation to the flood zone, a significant portion of the research area is classified under the categories of very low, low, and moderate. In summary, the region has a low index of susceptibility to flooding. And let's proceed into Forest Road Deformation Index. The volumetric measurements of excavation and embankment materials for the entire length of the road, together with the classification of corresponding deformations, were based on the 4 cm box range, which provides the highest amount of detail. The observed differences in surface deformation between the 3 cm, 4 cm, and 5 cm box range, figures 5A to C, do not demonstrate any significant variations concerning the geometry of the road, when compared with the on-site investigation seen in figure 5D. The results of the earthworks calculations are displayed in Table 2. Values designated as 1 signify fill volume, and values labeled as 2 indicate cut volume, whereas values over 55 cm are excluded and assigned the number 0. The pixel count was higher in fill volume than in cut volume. No considerable differences, up to 4.26%, between the cut and fill volumes were evidenced. We revisited our approach, utilizing the DM for the precise detection of deformations exclusively on the forest road surface, but this time with a higher classification precision of 0.001 cm, which resulted in the creation of 30 deformation classes. Figure 6 illustrates the deformations resulting from the vehicular movement and tire-induced pressure, figure 6A to C, as well as the visualization of water flow resulting from rainfall, figures 6D to F. Finally, we will proceed into discussion in the conclusion section. In our work, we employed a novel approach that involved two distinct classifications based on the DSM and the DEM, both implemented using the same technique. Furthermore, we conducted an analysis on 3 raster cells. The objective was to determine the optimal raster cell size based on the road surface topography. Such decision was supported by on-site investigations, considering the desired level of precision. Our research bears some relevance with previous studies who used UAV photogrammetry technology to detect soil surface deformation, implementing a polycopter UAV model and GCPs. However, in our case, precise measurements were possible in a much easier way by utilizing a fixed-wing UAV equipped with the RTK function, which eliminates the need for setting GCPs or validation points. And let's go to conclusions. In this study, we developed a novel approach for identifying and visualizing different types of surface deformation on a forest road. This approach aims to enhance the quality of the road, optimize its management, and obtain accurate data for performing technical operations and interventions on specific sections of the road. The aforementioned information will provide significant assistance to forest managers when constructing or maintaining trails and forest roads. Also, generation of flood risk maps is strongly suggested as a visualization tool of potential hazards. Thank you for your attention.
Many thanks, Evangelia, for her move from Greece. And the study of next speaker focus on genetic composition of breeding pine Pinus sylvestris population in Poland. And I'm pleased to invite Martina to make your oral presentation, please.
Good morning, everyone. My name is Martina, and today I want to tell you briefly about part of my PhD dissertation. And today I'm going to tell you about the genomic analysis of breeding Scots pine population in Poland. So, starting with significance of the species, obviously everyone of you knows how significant a species Scots pine is. And it is a foundation species of diverse forest ecosystems across whole Eurasia. And the range is the widest of all pine species, spanning from Scotland and Spain on the west till Pacific coast of Russia. And in Poland, as a main forming species, it's a great ecological, social, and commercial value, and it provides 60% of all timber production and covers nearly 70% of all forests in Poland. And the extensive range of occurrence is connected with high diversity of inhabited environments, and somehow it obligates Scots pine to develop a number of local phenotypic variants reflected in forms, types, ecotypes, and traits of physiological features. And right now, about 150 morphological variants has been distinguished within the species. And as you are aware of, and might heard yesterday from Professor Dederski, Scots pine, like many other coniferous species, now is under strong pressure from ongoing climate change and extreme weather conditions such as long drought episodes, floods, hurricanes lead to increased sensitivity to pathogens, pests, and parasites, and as a consequence of this, to higher mortality. And it was predicted that until 2070, pine will significantly lose its southern range of disturbance and will move northwards. And as a preliminary point, a word about Polish regionalization of seed material should be said. And here you can see a map of regionalization of Scots pine with currently 23 valid seed zones. And each of these seed zones has reserved seed stands, which are mature, well-formed stands with best phenotypic characteristic and capable for abundant seed production. And reserved seed stands are excluded from logging. And also, there are maternal trees, and as you can see here, a maternal tree has its own unique number, and it's registered and controlled by state forest. So if anyone is interested, it can be anytime checked on the state forest site, and each of the maternal trees are there. And the general aim of my study is about genetic variation. So now I'm gonna tell you some words about it, and what you should know here was the most important, that neutral variability does not influence the phenotype, and it's often used as a source of molecular markers in population genetics, and it reflects past demographic processes that have affected the species. And adaptive variability affects phenotype and results from natural selection and is under the influence of local environmental conditions. And the general aim of my study is to look at the patterns of neutral and potentially adaptive genetic variation of the most valuable ecotypes of Scots pine in Poland, which are managed under strict seed zones regime. Assess the genetic relationship between population influenced by historical and adaptive processes related to formation of local ecotypes, and provide data useful in the management of the existing genetic resources of the species in our country. And going to materials and methods, I have used 27 populations in Poland, which covers all of the seed zones, and it is about 840 trees in general. And all of these trees, which what's most important, are old trees, about 180 years old. And I have used mitochondrial DNA markers, 12 mitochondrial DNA regions, nuclear SNP markers, almost 40,000 markers, nuclear microsatellite markers, that was 16 polymorphic microsatellites. And here you can see a result, how does it look like in the lab, and this is a standard microtiter plate used for SNP analysis, and a sequencer, of course. So first results here, it's mitochondrial DNA. And as you can see, there are two most common haplotypes. First one is H1, the orange one, and it's present in every population, as well as H2, which is also, as you can see, in every population. What is most interesting about it, as you can see, they are quite not very closely related. There is 9-point mutation between them. And while I'm having 12 markers, this is a huge difference. And both of these haplotypes forms two main groups. And also the blue one haplotype belongs to phenoscandion mitotype, which is super interesting for me. And next, microsatellite results. As you can see here on this graph, Bayesian clustering using structure did not indicate any signature of population. Structure also principal coordinate analysis forms a mainly uniform distribution of genetics here, with one slight difference. Here the yellow is Menzeleche population, and it comes from mountainous region, which is the only unique population in our data set. And here, as you can see on this FST map, the dark point shows also Menzeleche. This it's still not a very big difference in genetic difference, but it's still the most differentiated from all of them. And SNPs, generally we can say that we observe high within population genetic diversity and low interpopulation genetic diversity. And here is the mental test, which shows correlation between genetic and geographic distances. As you can see, it's almost flat and not much correlation it shows. And also pairwise FST similarly, it shows only Menzeleche as the only distinct population out of these. And same with principal component analysis on SNPs. Just Menzeleche shows a little slight difference from all the other populations, which are grouped as one, basically. And conclusion. Most of the analyzed population represent homogeneous genetic group. Results indicate similar history for most of the studied population, with some genetic deviation found only in mountains. Genetic variability of the studied population is the result of admixture of population in different origin, and genetic data question the division of Scots Pine regions in Poland and contribute the ongoing debate about management of the Polish of the population and changing climate. Thank you so much.
Thanks a lot, Martina, for your very interesting presentation. The next young scientists study the relationship between functional and biochemical traits of model plant species in forest and temperature changes. So I pleased to invite to Beata for you. Will make an oral presentation, please.
Okay. Good morning. My name is Beata Czak, and I come from the University of Rzeszów, where during my PhD, and I'm researching impact of increased temperature for herbaceous forest plant, for example, yellow archangel. And during today's presentation, I would like to present some of my preliminary results, and I start. Let me start with why we choose the species for our research. On first, is common on deciduous forests almost all over Europe. and it occupies a significant part of the forest herbaceous plant layer, which plays an important role in the functioning forest ecosystem. For example, the proper circulation of water and elements. This slide shows general and main information for yellow archangel. Yellow archangel is an evergreen plant. Hammerfoot, they have leaves ovoid and covered with hairs. They have short and thin roots, shallowly rooted. They flowering from May to June, and they have yellow flowers. Thanks to the produced stolons, it also reproduces vegetatively. The objective of the work was analysis of the physiological and biochemical response of the yellow archangel to an increase in temperature. And the hypothesis is when environmental parameters change, a quick reaction will occur and a new equilibrium state will be established in the following days. We conduct our research in a specially breeding room, which we see in this photo. In this room, we have constant temperature, lighting, and controlled humidity. In this slide, I show the initial breeding from the photo in the photo A, and proper breeding in the photo B. Vegetative shoots of the yellow archangel were collected in our designated part of the oak hornbeam forest near Rzeszów. And these shoots were then divided into small parts and placed in sand as in photo A. After 3 weeks, they were transplanted into pots as in photo B. And about 300 individual plants grew 2 months in the breeding room under climatic conditions reflecting those in forest in Central Europe. Is 21 degrees in day and 30 degrees at night. And with a photo period of 60 hours of daylight and 8 hours of darkness. And these were the control condition. Control and test samples were collected randomly several times during the day and at night for the first 4 days and after a week. Today I will present the results for our selected hours of the day from 4 days and after a week. During the experiments, some of the analyses we made were chlorophyll fluorescence, performance index, relative water content, specific leaf area, dry matter content, and biochemical profiles of leaves obtained using infrared spectroscopy. ATR is a method of mid-infrared spectroscopy. It is used to study the structure of individual molecules and the composition of molecular mixtures. It allows for the identification of substance as well as the determination of chemical reaction paths and change occurring in the material. Different chemical structure produce different spectral fingerprint, and advantage is small amount of sample and often does not require special preparation, short research time, and very high precision. And now I move on to presenting the results. These 2 graphs show means changes and standard errors in the percentage of change in negatively correlated parameters, dry matter content and specific leaf area. The great difference can be observed after a week of elevated temperature. In the case of leaf dry matter content, it is increased by 22%, and specific leaf area is decreased by 22%. The height of the error bars suggested high variability of value during measurements, and average for all days from leaf dry matter content is 8.8%, and specific leaf area is decreased 2%. Next chart show means changes and standard errors in relative water content expressed as the difference between the control and the test sample. The great difference in the decrease in water content are observed after the first day of application of increased temperature. The high of the error bars suggested low data variability. In the following days, we can say that there has been acclimatization and or stabilization, and average for all days is decreased for 3%. This chart shows means changes and standard errors in fluorescence chlorophyll and performance index under the influence of increased temperature. The biggest difference can be observed in the case of performance index for all days, and average for all days is increased performance index by 47%, and fluorescence chlorophyll by 3%. Despite such a large increase in photosynthetic efficiency, the total chlorophyll content did not change, as can be seen in this chart. Now I will move on the results obtained with ATR. This example chart ATR show biochemical profiles of leaves yellow archangel. Blue is a control chart and red is a high temperature. I marked 4 peaks on the graph that showed a noticeable difference after the first day of the experiment. The marked peaks corresponded sequentially carbohydrates, next also carbohydrate, next pectin, and next also pectin. Based on the analysis loading plots, peaks were identified that determine the difference between the control sample and the test sample. For PC1 axis, explain peaks for carbohydrates, and PC2 axis for pectins. I marked these peaks on the previous slide. The first 2 PCA axes explain 47% and 23% of variance in the spectral data set. Non-metric multidimensional scaling based on the maximum peak absorbs values for a designated K-ATR range from the previous slide. C is a control group and T is a test group, and the number are the days of the experiment. The analysis showed that the sample were divided into 2 groups, control and experimental group. As you can see, the control group show greater diversity of feathers, which is visible in the form of a large circle. In conclusion, the yellow archangel is a good abandon guide for studying the effect of temperature changes. There was a quick reaction to the change in parameters on day 0, specifically for area, relative water content, performance index, and leaf biochemical compounds. On the following days of the study, the ecophysiological parameters constantly change. The increased temperatures is responsible for the increase in the dry matter content in the leaf, and the decrease and specific leaf area. Increased temperature causes an immediate increase in performance index already on day 0. ATR results confirm change in the metabolism of plants adapting to higher temperature contact with carbohydrates. Each of the analyzed parameters reacts in a different way to temperature change. It can be said that the plant's reaction is complex. And thank you for your attention.
Many thanks to Beata for your interesting and very important study. And the last topic of our third session is about variations of iron concentration in stem flow in the beech forest, Germany. And I'm pleased to invite to Tara. You will make an oral presentation, please.
Yeah. Hello. Good morning, everyone. My name is Tara Kaila. I come from University of Göttingen, Germany, and today I'm here to present my master thesis work for you. The topic is variation of iron concentrations in stem flow and through fall in a beech forest in Germany. This is my agenda for the presentation today. I want to briefly introduce about the topic that I'm going to present and explain about my research hypothesis and methodology and discuss the results and the discussion part and summarize the topic. Coming to the introduction, I would like to start with a short explanation of what is stem flow and through fall. I am sure most of you here know about it, but yeah, I just wanted to say what stem flow is. It's the intercepted rainwater that passes along the tree trunk towards the ground, whereas through fall is the rainwater that falls through the branches with or without getting in contact with the tree canopy. And why is it important to study about stem flow and through fall in forest? Because they play a huge role in nutrient cycling and water cycling in the forest ecosystems. And this is very important for the growth of the forest and also to sustain the forest in the long term. And due to this important role, we are interested in studying them, and we started our work with an objective to find the variations in the behavior of different ions in different partition fractions of rainfall. And here you can see the hydrological cycle of wooded ecosystems where stem flow through fall and other precipitation fractions are shown here. Before going into presenting my actual work, I wanted to tell about our work, which is really a long experiment. It started in 2017, and the first half of 2017 year was mostly for planning the work and just brainstorming about the objectives and what to do with it. And the next half was mostly for setting up of the infrastructure and starting of the experiment. In 2018, research and development work was done, mostly for collecting the data, rectifying errors, which was within the instruments that were set up in the field. And from 2019 to 21, very extensive sample collection and analysis was done. And in 2022, I started my master's thesis work for analyzing the data from the previous years and also in 2022. And I continued the data analysis with a focus on ion concentrations of stem flow through fall and rainfall samples. And in 2023, I submitted my master thesis. Our objective for this work, for my master thesis, for this work was to see how different ions in different concentrations was varying in these three partitions of rainfall. So to achieve this objective, we hypothesized that the larger the tree size, the higher the ion concentrations in the stem flow, and the ion concentrations of stem flow will be higher compared to through fall and rainfall concentrations. And there will be variations in stem flow through fall ion concentrations between different climatic seasons. The main factors that we considered for my work are volumes and ion concentrations. The ions such as potassium, calcium, magnesium, sulfur, sodium, phosphorus, aluminium, manganese, iron, and zinc of stem flow through fall and rainfall. And we also considered meteorological seasons or climatic seasons for the third hypothesis. This is the experimental site. The experimental site is located towards the northern part of Germany in a place called Ebergotzen. And in the experimental sites, there were many trees, out of which I chose 12 trees for stem flow and 4 trees for through fall analysis. Within the 12 stem flow trees, I have divided them into big trees, small trees, and medium trees, and based on their diameter at breast height. The big trees are the trees which have more than 40 centimeters in diameter, and medium trees are 20 to 40 centimeters, and small trees are less than 20 centimeters. My experimental approach here was the setup was done, everything was done in the previous years, and the volumes data was collected using data loggers, which is automatically collected every 15 minutes intervals. And this 15-minute interval data is automatically in the data logger, which can be integrated into the SQL database once a day, and this is extracted through a R script which was done especially for this experiment, which is called Ebergotzen. And the stem fall measuring resolutions with these tipping buckets here was 0.5 liters for big trees and medium trees, and 2.25 liters for small trees. And this collecting vessels is used for through fall water collection. Ion concentrations was done on bi-weekly basis. For laboratory analysis, which was done by our department lab technicians, we used ICP-OES, inductively coupled plasma atomic emission spectroscopy. Statistical analysis was done by me. I used MS Excel for doing the data organization and the previous data setup, and our program version 4.2.1 was used to do the analysis using the following packages. For the data analysis, I did mostly ANOVA and regression analysis. Coming to the results, here are the results which showed that the first hypothesis showing the differences between ion concentrations between big, medium, and small trees. It it wasn't clear pattern that it wasn't shown that big trees were higher in concentrations and small trees did not show any differences. We hypothesized in the other way, but this can be attributed that because of our methodology that we used, where we collected the stem flow volumes in a similar way for all the types of trees, we couldn't find any differences in the ion concentrations in any of the ions. Here comes the differences in stem flow, true fall, and rainfall. Our results showed that stem flow concentrations are significantly higher than true fall and rainfall concentrations for almost all the ions. Our results were in accordance with Johnson and Lehman studies which were done in beech forests, and this is already expected. And we attributed this to the canopy exchange and leaching of ions from the stem flow into the rainwater, and also due to high residence time in case of stem flow compared to true fall. And here is the volumes in stem flow, true fall, and rainfall in each year. It wasn't as expected, and it is not comparable to other studies, but there are also studies which had similar reports. In our case, we had way lesser stem flow compared to other studies, which was averaging around 7%. But there are several other studies in beech forest which showed up to 20%. This was due to the fact that we did on biweekly intervals instead of doing on a rain event basis. Here is the stem flow, true fall, ion concentrations in different climatic seasons. The concentrations were found to be higher in summer and autumn compared to winter and spring seasons. This was attributed due to the leaf senescence phase during these seasons. During this time, the mobility of nutrients is extremely high compared to other seasons. Thus, this can lead to a mobility of nutrients into the stem flow, causing high leaching. And in summer season, there will be high exposure of the leaf area, and also the contact surface for the leaching process will be higher, which can lead to more stem flow concentrations. I want to summarize that the effect of tree size was not evident for most of the ion concentrations, which can be due to the methodology that we have chosen. And the volumes of stem flow and true fall is not comparable to other studies, even though there are some studies which proved our research to be true, but this could be rectified by doing the volume analysis on event basis. And the seasonal impact on ion concentrations was shown that autumn and summer ion concentrations were greater than spring and compared to winter ion concentrations. These are some of the references, and I thank you very much for your attention.
Many thanks to Tara for your presentation from Germany, and many thanks to all speakers. Let's go to discussion panel. Who want to ask question to our speakers? Who want to ask question to Christina about your study, bryophytes? Please.
Yeah, thank you for your bryophyte talk. It was very nice, and I'm happy that our results are quite similar. I have a question because you told us about that bryophyte diversity depends on presence of deadwood in forest, and I have a question: did you determine the species of dead wood, or you just treat them as just dead wood? That's all.
Thank you for the question. It is a good question, and maybe we should have tried to determine the species, but as it was already 2 decades after the wind event, it was quite difficult to determine the species after that much time. Some of the logs were already in quite a late stage of decay, so we unfortunately couldn't identify them anymore. But it is a very good question, and actually we have in mind to do a study when we can involve such effect. So thank you for the question.
Many thanks for your answer, Christina. Who want to ask question, please?
Thank you for your interesting presentation on bryophyte. My question is, in one of your results, you showed that in moderately damaged plots, there is more diversity of bryophyte when compared to heavily damaged plots. So I want to know, can we say like wind disturbance help to support bryophyte diversity? And secondly, what could be the implication? Like, what caused the difference in heavily damaged plot compared to moderately damaged plot?
So thank you for the question. Yes, you understood it correctly. Moderately damaged plots hosted greatest species richness, and we suppose that the reason behind it is because the heterogeneity of the environmental conditions is highest in those plots. And as we know, bryophytes are a bit sensitive towards moisture content, and moderately damaged plots have remained quite intact canopies, especially 2 decades after the event, when already the regeneration has started to grow quite nice. So they do not desiccate, and they feel very well in these conditions. And probably this is the reason why these plots had also the highest number of unique species present. And as we know, wind disturbances have already increased in the frequency and the intensity, and they are expected to increase in the future as well due to climate change. So the disturbance management is very important in this perspective. I hope I answered your question. Thank you.
Many thanks for your answer. Anyone else want to ask a question to Christina? Okay, I have one question. What do you think will be confirmed your conclusion in other environmental conditions such as tropical forests?
That is a good question. I suppose that it can be true, but bryophytes are quite different in terms of composition. In temperate and boreal forest, we have a higher species richness of mosses, but in tropical forests, there are a high diversity of liverworts, and liverworts are known to respond a bit differently to environmental factors. So I think that there are differences, but I think that the main conclusions are probably the same.
Thank you very much for your answer. And what question do you have to Martina about your study? Who want to ask question about genetic compositions?
I saw on the map that metotypes in Poland are quite, maybe not evenly, but are quite mixed in all the stands. Only this one stand, Międzylesie, was different. And I want to ask whether it's Poland the melting pot for the different lineages of Scots pine, or maybe the situation is similar also in other part of European range of species. What it looks like. It is a difference in, between Poland and rest of the species range or not?
Thank you. That was a good question. Actually, I was about to tell it. I'm sorry, I didn't. And yes, we can say that Poland is quite of a melting point for diversity in Europe. And it's the only place actually on map that probably different lineages of a region have mixed in Poland, and that's why it looks so colorful on the map that, as you could see, it's not like this in the other parts of Europe. For example, in Venoscandia in Finland, there's one main mitotype, and in Southern Europe is another main mitotype, which corresponds to our main. So probably Poland was on the way of the diversity. I'm sorry, you know what I mean. Thank you.
Thank you, Martina, for your presentation. My question is related to one of your conclusions, where you showed that studied populations show homogeneity, like they're in one of your conclusions. So I want to know, like, okay, if they are quite similar, what could be the reason? Is it due to the method of propagation of this Scott pine? Like, is it due to propagation by stem cutting, or because that could bring about the homogeneity when compared to propagation by seed, which can bring about variability. So that is the first question. Then the second question is, since they studied allotype, they are quite similar. So is it that the future of Scott pine in Poland is really scaring, because could it be that we may find it difficult to see plant species that will show adaptation to future climate scenarios?
Thank you for your question. So answering the first one, the problem is that Scots pine is a wind-pollinated species. And it looks like in the whole range that the whole range is almost the same, and the differentiation is about 2% on the whole range. And in Poland on SNPs it's less than 1% even. And corresponding to your second question, yes, but Poland is still most diverse in Europe, as I can say. So I see hope in this mitochondrial diversity, that maybe thanks to this huge diversion that we have in mitochondria, which is mitochondria is maternally inherited. So it's in the seeds. So we see the, let's say, old maternal mitochondrial DNA that was first here. So this look has some kind of hope that maybe our Polish Scots pine could get along with this climate change and probably be better suited than the northern one, where it's just mainly one mitotype. I hope I answered.
Thank you for your answer to question from participants. Anyone else want to ask question? I have a question. What's your opinion? Can the genetic heterogeneity of Pinus saludistris population be caused by impact, for example, falling of different intensity or soil moisture?
I'm not sure if I understand. You said about moisture?
Soil moisture and falling of different intensity.
I didn't thought about this aspect because, as you could see, I'm mostly. Well, I think that yes, because Scots pine is a highly plasticity species, so I think it's connected with soil moisture and so on. And I would say that the differentiation is connected with microclimatic conditions that we have in Poland. And as you could see, for example, Eastern Poland was slightly different than Western, which is maybe the reason why there is more wet soil in the east. Is that the answer?
Thank you for your answer. Does anyone want to bring up anything further to Martina? Okay. Thank you very much for your answer. And what question do you have to Beata about your study about Galeopdolon luteum in Poland? Who want to ask question? Please.
Thank you for your presentation. I would like to ask you about the type of material. What do you need to this analysis? What part of plants? Can you tell me?
We collect leaves for our plants. 5. And one ATR result is one from one leaves.
Many thanks for your answer, and we have one question.
Thank you for your presentation. Just a quick question, because I noticed that you use for your model plant. What makes it different? Because for so many studies, they usually use Arabidopsis. So I don't know what makes your model plant unique or different for your specific study when compared to those studies that have used Arabidopsis to look at temperature variation for plants.
Can you repeat shortly?
Okay, what I'm saying is that from most studies, they use Arabidopsis thaliana to look at effect of temperature, effect of carbon dioxide, carbon monoxide on plant physiology. Like they use this particular plant species. So for your own study, you use a different plant, which is the yellow ash angle, right? So I want to know, like, why do you choose this particular species compared to you not using Arabidopsis? So.
We use yellow aranja because this species is forest species and they are a significant part of forest, and the results results for temperature So wyniki, które uzyskamy na tej roślinie, która jest typowo leśną rośliną, będą ważne ogólnie dla funkcjonowania całego ekosystemu leśnego, a Arabidopsis to jest taka typowo roślina zielna, która nie rośnie w lasach, a dla nas tak ważnym, tak. Chciałam więcej powiedzieć i nie wyszło.
Thank you for your international answer, bilanguage answer. Anyone else want to ask a question to Beata? I have one question. Can I ask? The appropriate relationship between functional and biochemical traits of Galeopdalon luteum was established in your study. And what do you think? Will such links be confirmed for other species of this genus, for example, Galeopdalon montanum in Poland? What's your opinion?
I think
Discussion panel. To be or not to be?
not.
Okay. Okay. Thank you very much. And the last study was about variations of iron concentration in stem flow in beech forest. Who want to ask question to Tara about your study? Please. Because I have one question too. Please.
Yeah. So you were working on a European beech, right? And only this one species. Okay, so I have a question. Do you think that your results can be extrapolated to others for three species of temperature forests, or is it only for European beech?
Yeah, these results, I think it can be differed for other species because European beech is very different in architecture. It has a very wide architecture, and for this reason, it has high stem flow values compared to other trees, which has lower stem flows. So I think this cannot be the same with other tree species.
Thank you for your presentation. I want to know if you do the same study, but in the area where the urbanization is high, around the towns or around the mine activities, do you think that the result can change or not?
Yeah, I think it can change because the area where we did was not urban area. It was mostly forest surrounded by a very small village. And with urbanization, I think the air concentration, like the concentrations of elements within the air and within that, and microenvironment changes which can result in different concentrations within this study.
Many thanks for your answer. Anyone else want to ask question to Tara? Please, Professor Marcin Petras.
I want to ask about the management system that you use in Germany. because in different countries we have different system management of beech forest. In Poland we have quite intensive system with a lot of cuttings in 5 to 10 years periods. For example, in Italy they have very limited cuttings, so maybe you are between Poland and Italy.
I'm not quite sure about this question, but the forest that our experimental site was in, it was very well managed with the by the Department of Forestry faculty in University of Göttingen, and there was no cuttings done. It's quite old, like more than 150 years old forest.
Thank you very much. Anyone else? Okay, I have one question. Your method take into account the size of trees, in particular diameter and height. Have you investigated the age of trees to make the valid conclusion? Or classes, age classes?
Yeah, for my study, no, but the other studies by my teammates, they are considering also the age, and also I think the size of the trees also correlates a little bit with the age. The more age it has, the more diameter in general. But I am not sure if it is the same with every tree. And in my case, I did not consider the age or any age class.
Thank you very much. I don't have any question. Thank you to all speakers, to all participants for work of our third session. Thank you very much. And now we have a coffee break. Thank you very much for your work.
Good morning, everybody. I would like to welcome you on this session. My name is Teresa Chazubska-Przybył. I'm very pleasure that I can host to this session. And now we have 4 speakers today, 2 from abroad research centers and 1 from Poland. And I have I have a request to all speakers to keep your time, and now we can start. The first person is Quentin Gaidos from Belgium University, yes, and he will present the topic unraveling below-ground interaction in occum stands, the roles of root anastomosis and mycorrhizal association. The topic is.
Yeah, thank you. Thank you. I just put it a bit higher. Yeah. Thank you, everybody, for being here today. And thanks as well to the organizing committee for giving me the opportunity to talk about my thesis topic today. So we'll talk about Okume. Okumea kleiniana Pierre of the Burseraceae family, which is the most harvested timber species in Central Africa, despite the fact that it only grows in the 13 most western part of the Congo Basin, which is the second largest tropical forest in the world after Amazonia. With 3 other species, it accounts for almost 70% of the overall production in the whole region. It's a pioneering, light-demanding species that is dispersed by its winged seeds that you can see on the left to colonize open environments such as savannas, wood sides, fallow lands, or opening gaps. Yeah, it wasn't me, I think. So when it grows, it becomes monospecific to monodominant dense stands, and then it becomes scarcer in old-growth mixed forests. Okume is also famous for its root anastomosis, also called root grafts, which are basically root connection between neighboring trees of the same species or not. It's been first discovered during the 20th century when foresters in the region perform, as usual, thinning operations. Well, they came back years later and they could find that the stumps remained alive even after a few years. About that, Leroy de Val in 1973 wanted to check if it was the fact of those root grafts or not. So he excavated 7 stands in the forest to make sure that the trees and the stumps were actually connected or not, and he mapped it. But he didn't prove if it was only mechanical merging of the roots or physiological. So what's the matter about Okume? Well, there is a rarefaction of open environment suitable habitats for it. First, because of the natural aging of the forest that's closed up over time because of a lack of natural perturbations. Second, because historically, local population used to be nomadic. So they came to a new place, cut the forest, cultivate their fields, harvest it, of course, and then left. Then it left open environments really interesting for pioneering timber species, such as Okume, which is not the case now. Third reason, in certified logging companies, they only harvest 1 to 3 trees per hectare, and by using reduced impact logging techniques, which leads to too seldom and too small opening gaps, averaging 300 square meter in size, which is too small for new population to establish. Obviously, foresters extensively planted the tree in order to try to refill the stock, but it gave mixed results, mainly due to a poor knowledge of the species ecology. So there is a lack of regeneration of the species and a decline in its productivity. It's even classified as vulnerable by the IUCN even though it is debatable. The main risk about that is a conversion by the countries in which it grows to more cost-effective land uses, such as agribusinesses like palm oil plantations. In this context, my thesis aims to identify optimal conditions and ecological mechanism for the establishment of new Okume populations. We aim 3 objectives. The first one is to quantify the optimal light condition for the establishment on the seedlings, but I won't focus on that today. I prefer to focus on the 2 other objectives. The second one here, we started back from the observation of the wood graft about which we talked before. And we tried to measure the impact on stand structuration and growth of the trees by trying to ask 2 questions, but I'll talk about it later. And of course, we couldn't study below-ground interaction between okumes without studying mycorrhizal network and more generally fungi, root-associated fungi that should also have a role in the stand dynamics. Now I'll present the methods and the preliminary results of it. First, about the root graft experiment, we made use of Dendrology, which is basically the study of growth rings over time. For that, we started from the same protocol as Laura Deval by excavating 3 stands in Gabon, and we collected trunk sections, root sections, and root grafts from it. How did we do that? Well, first, we inventoried 3 stands in Gabon. Then we clear-cut it, and after that, we drove a fire truck in the forest in order to excavate the root system of the stands, thanks to the fire hose. After that, we've been able to collect our samples. And for now, we are sending the samples in order to perform the ring analysis later, but it's still ongoing, unfortunately. When it's going to be done, we will want to answer our main hypothesis, which is, do root anastomosis boost dominant tree growth or not? To expose the goal of it, I'll take the example of Pinus contorta on the right, Okumea is on the left, because it's easier to observe on Pinus contorta. So here you have a root graft formed by 2 different roots belonging to 2 different trees. You can see that the root graft is 5 years old because there are 5 common rings. Well, we expect that the dominant tree from which this root belongs would have its 5 last growth rings wider than the previous one, thanks to the benefit of an extended root system from the root graft. And in exchange, it keeps the suppressed tree on the left alive from the grafts. Besides that, we also performed another experiment because, as I told you before, Leroy Deval in 1973, he checked for the merging of the roots, but he didn't check if it was only mechanical or also functional, meaning sap redistribution between trees. So we looked for groups of okume in the forest. We cut one, and we built on top of it a tank that we filled with fuchsic acid, pink dye. And we came back three weeks later in order to see that the dyed sap had been transferred to a neighboring tree, proving that there is indeed a redistribution between trees. About mycorrhiza and root-associated fungi in general, we also wanted to check that in order to see if it impacted stand dynamics more or less than root grafts. To do so, we collected on more than 100 trees in Gabon fine roots from young and old forests, from dominant and suppressed tree. And as you can see on the right, those are okume fine roots. Their morphology is pretty atypical, and you can see outgrowths, pear and club-shaped, which which really looks like ectomycorrhizal associations that have never been discovered about this family. So we checked about that, and more especially because a lot of other studies linked the monodominance of stands with ectomycorrhizal associations. So first thing that we've done is stain the roots that we collected and observe them microscopically. But unfortunately, we cannot see any mycorrhizal features, such as we saw yesterday for ectomycorrhiza, heartugnet or mantles, or even arbuscules from arbuscular mycorrhiza. It might be due to seasonality, the period when we collected the samples, but we cannot make sure about it. So we stopped this one, and we opted for molecular methods using NGS Illumina sequencing. First, we had to adapt our own protocol because it's been more complex than expected to extract and amplify the DNA. But when it's been done, we amplified the ITS2 region, which is fungi-specific. And then we obtained our OTUs, and we assigned it taxonomically with UNITE and ecologically with Fungil. First three results about it. First one, ectomycorrhizal fungi do not explain monodominance in the stands, simply because there are almost no ectomycorrhiza. Over 13,000 OTUs, 1/4 have been classified as symbiotrophics, and from them, only 36 are being classified as ectomycorrhiza. So it would be more the fact of contaminants or contact mycorrhiza. But there is a persistent presence of EMF in every samples, mostly Glomus spp, which is not really surprising because they are really widespread in the world, but still it needed to be confirmed for Okume because nothing has been done for these three about it before. Third, preliminary results. The stand stratification, meaning dominated versus suppressed stand, doesn't seem to be impacted by root-associated fungi, regardless of ecological guild, meaning semiotrophic, pathotrophic, or saprotrophic. Either for alpha diversity, meaning species richness, or for beta diversity, there are no significant differences among every indexes. So no impact of mycorrhiza and root-associated fungi in general would mean that it would be more about root grafts. And despite the lack of positive results about it, we are the first that are characterizing the microbiota of Okume that has never been done before. So as a conclusion, so far, we learned that suppressed tree can survive with limited light, despite the fact that they are supposed to be pioneering light-demanding species, thanks to the research sharing from canopy trees and through root grafts. The monodominance isn't explained by ectomycorrhizal associations because there are no ectomycorrhiza, but there are AMF, arbuscular. That's what I've just said before. No difference among social status explained by the fungi community. But for the first time, at least, we characterize it. One of the most interesting hypotheses is, do root anastomosis boost dominant tree growth or not? I don't have the answer yet, so stay tuned about it. And all of that would imply a better understanding of Okume population ecology in order to re-establish stable, productive populations while preserving biodiversity. Thank you.
Thank you, Quintin. You are on time, so everything is okay. Maybe one question from after. Okay, so now we will the possibility to listen to the presentation of Hannah Fuchs, Mrs. Hannah Fuchs from our Institute. And the topic of her presentation is redox dynamics in seeds of woody species, unraveling adaptation strategies for different seed categories. So,
thank you for the introduction. I am Hanna. I work in the Department of Developmental Biology in the Institute of Dendrology, Polish Academy of Sciences. And today I would like to share with you our results of comparative analysis of thioredoxin target proteins in different seed categories of woody species. So I don't think I have to explain why seeds are so important for ecosystems and our economy. This topic was well explained yesterday by Professor Eva Kalemba. So I would like to say a few words about models in molecular studies of woody plant seeds. As you may know, molecular biology is sometimes very tricky, so it's very difficult to manage research on a large scale. We have to simplify things, and we use models such as Arabidopsis or rice or peas. But in woody plants, we propose three species that are very good models for study seed categories. So the first one is sycamore maple, and the second one is Norway maple. They both are the part of genus Acer. They are closely related, and their distribution is very similar. Our third model is European beech, and maybe it's not very closely related to the other two, but it's very important forest-forming species in Poland and Europe, so it's a very viable source in the research. All of these three species produce seeds of different categories based on their desiccation sensitivity. Sycamore maple produces sensitive to drying seeds to humidity below 27% and conventional storage conditions. They remain metabolically active, therefore they are categorized as recalcitrant. Norway maple seeds tolerate drying to humidity below 7% and storage at -10 degrees Celsius, and they silence their metabolic activity, so they are categorized as orthodox. And last but not least, European beech produces seeds that during storage lose their viability relatively quickly compared to orthodox seeds, therefore they are intermediate. So our department is interested in those species for many, many years. So the literature available in the databases is very rich. So the physiology is quite well known right now. So these features makes those species available models for study seed physiology. Okay, moving on. What are the factors necessary for development seed tolerance to drying and storage? And the answer is that they're mainly accumulation of spare proteins and fatty acids, accumulation of protective proteins, impermeable seed coat, silencing the metabolism, and active redox and antioxidant system. And all of these factors are under control of ABA-regulated gene expression. We are mostly interested in active redox and antioxidant system, in particular in thioredoxins, which are proteins that are master activity regulators of other proteins. They are present, I think it's safe to say, in all living organisms. They are involved in at least 12 mitochondrial processes, from processes related to energy production and metabolism to protein synthesis, stress responses, and communication with other organelles. They also act as chaperones under oxidative stress in mitochondria. Despite their significant importance for cell function, there is still a huge gap regarding how the thioredoxin system influences seed physiology. So we asked ourselves, will the target proteins of thioredoxins vary in seeds of different categories? And to answer this question, we decided to identify the targets in the seed categories of our model organisms. And to do that, we had to cope with some difficulty, which is the ability of thyroidoxin to release its target protein within moments. It doesn't create a stable complex. And this is due to the motif that contains two cysteines, and those cysteines are responsible for creating the disulfide bridges between the thyroidoxin and the target protein. So how did we manage to do this? How did we manage to create the stable complex that could that then we could identify our protein? We changed the second cysteine in the amino acid sequence from cysteine to serine, and therefore the thyroidoxin lost the ability to release the protein, target protein, created a stable complex that could be further used in affinity chromatography. I will briefly explain the method. Maybe not everybody is familiar with it. So we had a bead with a ligand, which is our thyroidoxin, our mutated thyroidoxin, and then we add protein extract. The target proteins create a complex with our ligand. In the meantime, we remove remaining proteins. Then we release the complex from the beads. And then we have to break the complex. So we added the DTT to break the disulfide bridges. The next step was adding the anti-thyroidoxin H1 antibodies, so we could get rid of thyroidoxin. The target proteins remained, and then we could use them in the liquid chromatography and mass spectrometry. Thanks to this method, we identified 238 proteins in sycamore, 182 proteins in European beech, and 69 proteins in Norway maple. So as you can see, the first difference is in the size of the proteome. Then we classified our proteins based on the function they are on their function, and as you can see, the metabolism and genetic processing proteins predominate. On this chart, you can see a similar result, but in the percentage terms. So the sycamore acer was characterized by the metabolism proteins predomination with 38%, and the next were 34% of genetic information processing proteins. In the case of European beech, it was quite slightly different. Genetic information processing proteins predominated with 42%, and the metabolism proteins stands for 29%. Similarly, in Norway maple, the genetic information processing proteins predominated with 48%, and the next was metabolism with 26%. We don't have time to discuss all the proteins, so I will focus on those we thought might be associated with adaptation strategies. So in sycamore, the ATP synthesis is very visible here. As you can see, we have here almost all subunits of ATPase synthase. Also, the TCA cycle showed some representation. We had the malate dehydrogenase, exoglutaride dehydrogenase, and the other important function was stress response. We had some LIA proteins, dehydrins, and superoxide dismutase. On the other hand, the proteome of Norway maple showed the silencing of the metabolism. We found only one ATP synthase subunit, which is more interesting. This subunit was absent in the sycamore proteome. And the transcription and translation control proteins predominated in the proteome of Norway maple. In European beech, the genetic information processing also predominated with DNA replication, transcription, translation control, but we also found some proteins related with stress response. And what was characteristic for European beech, it were proteins that are involved in removing misformed or damaged proteins from cells, like polyubiquitins or proteasome subunits. So these proteome profiles for each category reflects, in our opinion, the molecular adaptations and strategies used by the seeds of the category studied to meet their distinct physiological requirements. The greater number of specific proteins in recalcitrant seeds may indicate the need of specialized molecular mechanism to overcome storage stresses, whereas relatively lower number of specific proteins in orthodox and intermediate seeds suggest a more conservative set of molecular processes optimized for successful seedling germination. So in conclusion, Sycamore maple is characterized by a greater number of pterodoxin target proteins than Norway maple and European beech, and the profile of these proteins indicates higher metabolic activity. Beech is characterized by an intermediate number of target proteins. Their profile indicates that beech seeds invest energy in removing misformed or damaged proteins from cells and control the level of specific proteins. And the protein profile of Norway maple indicates a reduction in metabolic activity and investments in the control of genetic information processing, which connects it with beech seeds. Furthermore, our research corresponds to the latest research, which indicates an increased level of redox regulation activity in sycamore maple seeds. So as it's often in science, now we have more questions than answers. So what is the role of pterodoxin-related system in acquiring desiccation-tolerant seeds? Is metabolic regulation crucial in this system? Does this system undergo dynamic changes during seed maturation? What causes the target proteins of pterodoxins to vary depending on the seed category? And the most important question, can we influence this system in order to obtain seeds more resistant to desiccation and long-term storage? So these are the questions that we would like to answer in the future. I would like to thank all the co-authors, especially Antonia Serrato, Paola Vargas, and Mariam Sarve from Zaidin Experimental Station in Granada, Spain, where we conducted the identification of the target proteins. Thank you very much for the attention.
Thank you, Hania, for your interesting presentations. So now we ask Mihail Brodsky from University of Tartu in Estonia, and the topic of the presentation is impact of elevated air humidity on the endophytic fungal communities in foliage of silver birch, Betula pendula, and Norway spruce, Pisa abies. The floor is yours.
Greetings, my name is Mihail and I come from Tartu. And since my topic was already introduced briefly, you might be wondering why elevated air humidity. Well, the climatic changes predicted for Estonia and other northern states state that the precipitation and with that the air humidity, the average relative air humidity will increase in the near future. And as well as that, some extreme weather events will become more common. That includes, for example, drought, storms, and as well as waterlogging in the spring. And the length of waterlogging is also expected to increase. So the effects of such change on forest ecosystems is what we are trying to predict. Partially on natural forest, partially on our experimental site. And for our study, of course, we chose spruce and birch because those two tree species make up almost 50% of all forest stock in Estonia. So first of all, about silver birch. It's a deciduous pioneer species. It's usually relatively resistant to all pathogens except for some leaf rusts, but it can be really susceptible to rot, to wood rot. And the best known rot-causing fungus is Inonotus obliquus. You might also know it by the name of chaga. And another well-known pathogen of birch is Taphrina betulina, which can be seen on this picture. And do not confuse that with the mistletoes growing outside. And silver birch is also considered rather resilient to climate change. And I've heard that they are planning to increase its presence in France, for example, to combat the effect of warming on the forests, but I'm not too sure about that. So the second tree is spruce. It's one of the most commercially important trees in Europe, and the biggest threat to it is Heterobasidion, which causes the root bud rot, and it causes up to 1 billion euros damage every year in forests. And another threat is not fungus, but it's an insect called Ips typographus. It eats everything under the bark of the spruce. It's larva eat it. And so Norway spruce is rather vulnerable to drought stress and excess heat, as it seems. So it's declining in the southern parts of its areal, and is predicted, as I think you saw yesterday, to move rather far up north with its area. So a bit on the foliar fungi. They are a complicated bunch. They often exhibit a very variable lifestyle. Even one species of fungus can be a pathogen, a saprotroph, and an endophyte all at once, although usually it would be a different tree species. They also, a lot of saprotrophic fungi grow in the leaves while the leaves are rather young, and rather after the leaf senescence, they tend to switch to saprotrophic lifestyle to get an advantage in competition for the nutrients that are left over in the leaf after it dies. Usually the foliar fungi are very low with host specificity. They often grow on many different plants and very unrelated plants as well. So they can also be really important for plant health, but how? That's another difficult question, because obviously there are some pathogens that can cause some rapid defoliation of a tree, but then again there are some other fungi that fight against those pathogens, and some other fungi that just promote tree growth, and so on. So they can also be an important marker for tree health when we are analyzing it, because they are partially overlapping with fungi in wood, and fungi in wood can, when they are pathogenic, they can be way more dangerous than the foliar fungi. For example, on the ash trees, the Hymenoscypus fraxineus, which is rather famous these days, it grows both in the wood and on the leaves. And so another aspect for those fungi is that the tree foliage is a relatively hostile environment for them, and you would find it difficult to survive on a leaf because it's such an unstable environment. It consistently gets thrown around by all the different atmospheric disturbances, as well as for fungus. The plant itself can inhibit its growth. the plant can fight against it with different strategies. Some are capable of producing some antifungal compounds, and the most common things on plant leaves are wax cuticle, which inhibits the entrance of fungi through the stomata. You can see those round holes are stomata, and those are covered with wax, partially to protect from the fungi and partially to protect from excess water loss. And those slim lines, those are the fungal hyphae. So our experimental area, free air humidity manipulation in South Estonia, we have over there 3 different treatments. One, of course, is control. One is air humidification, we mark it with blue, and one is soil irrigation, mark it with yellow. It's an oxalis site-type forest, which is one of the most common forest types in Estonia. Makes up close to 20% of all forests, I think, and it's named, of course, after this oxalis plant, which is rather tasty. So on our experimental treatment area, we imitate the predicted rise in precipitation, which I was talking about earlier. And for our study, we made 3 rounds, actually 6 rounds of sampling every year 2 times from 2020 to 2022. And we collected each time about 100 samples of birch leaves and 100 samples of spruce needles. And with those we later sequenced the DNA we have inside them. So from that, let's jump to results immediately. So this is the principal component analysis graphic for the birch leaf communities in yes. So with red and orange, we mark the control and soil irrigation treatment, and with green, the air humidification. And as we expected, the air humidification modulated the fungal communities quite a bit in a way that's somewhat difficult to describe right now, but Permanova says that the experimental treatment accounted for about 6.5% of all variability in the fungal communities. The sampling year and sampling month, however, accounted for a bit of a larger part in variability at 10%. And what you can see on this graphic is that the sampling months we sampled in July and September are also quite different from each other. We could make a line which goes like this about, and on the lower part we would have all the July samples, and on the higher and right part we would have all the September samples. Another thing you can see is the yearly variation. 2020 and 2022 are rather close to each other and make up the bulk of this part, and 2021 makes up all the extremes as well as part here. So in spruce needle communities, the picture is slightly different. We found here about 1,200 taxonomic units in total. And we found a similar picture that the treatment, experimental treatment, accounts for about 6% of total variation in the fungal communities. And the sampling year accounts for also 6%. And With the yearly picture, it would be somewhat similar to Birch, with 2021 making up all of the extremes and 2020 and 22 making up bulk of here. And sampling month on spruce is rather less important at only 2%. That is because, of course, the spruce is a conifer and they don't throw away their needles at the first year. And so they can't let too many fungi enter their foliage immediately. That we skip. And onto the overall taxonomic richness in birch, we could see that in the H plots, the humidification plots, the taxonomic richness is consistently lower. And the taxonomic richness, of course, is way higher in September than in July because, well, the immunity of the leaves drops and fungi just start colonizing it very fast as the senescence is approaching. In Norway spruce, the air humidification treatments, the taxonomic richness is lowest in July, and in September there is no significant difference between treatments really. And the difference between September and July is way lower, as I said before. So, indicator species. On birch, for example, several members of the Taphrina genus, which I mentioned earlier, disappeared with air humidification, as well as several Romularia and Exobasidium species, which are pathogens. And for example, Vishnia kazuma, which is an endophyte that later becomes a saprotroph, shows a high preference for H plots. About most abundant taxa a little bit. The most widespread on both trees are some Cladosporidium species and Aureobasidium pullulans, which can be seen here, which is a dominant yeast-like fungus on many different plant species, and can be used sometimes for biocontrol of pathogens. So what do I conclude? The lower species richness in H plots in birch leaves in September especially is possibly correlated with the later senescence that is caused by our air humidification treatment. And negative relationship between yearly precipitation and fungal species richness, for example, has been detected in Lithuania and before in spruce needles. However, not in spruce roots or the adjacent soil, for example. So we kind of corroborate with that results. And basically most of the variation in communities between years is caused by the natural fluctuations and partly because of the change in technology that they used for the sequencing, but I can't explain it now. So thank you.
Thank you, Michael, for sharing your results. And now we will ask Mrs. Olga from University of Environmental and Life Sciences in Wrocław for giving your presentation.
Okay, so thank you so much for the introduction. I would like to say something about the common beach, European beach, and what happened with the temperature gradient in Poland. So as you know and as you feel, global warming and global changing, it's happening right now. And the same feel the trees. as many beech trees sites are becoming less variable. Higher temperature, less rainy days, and more sunny day in the same moment have made beech trees more susceptible to pathogens. And I decided to check how temperature gradient can depend to genetic variability in the European beech. So for material methods, I decided to separate it for a free section, like in the field, in the lab, and on the computer. So in the field, I think it's the most interesting and most funny. I decided to make 9 separate region with the different temperature where the variated by 0.7 Celsius degrees. And there you see the map with this gradient. The red is the warmest and the blue or purple is the coldest area. So from here you see the south part of Poland, and from here the most important is this one point, GBO. No spoilers, but please remember this one point. So in the field I collected from this one point temperature area, I collected 5 samples from mature trees and 5 samples from young trees. The samples, it was 3 or 6 leaves. And with the leaves, after in the lab, I isolate the DNA using the Sherlock kit. And after send it to sequencing by a DART-seq method. With the results, I did some analysis on using the RStudio, like PCA, RDA, and others, using these packages and others also. So come to the results. Here I decided to 4 sections. So go in the first, genetic variability. Here you see 2 plots, 2 graphs. On the first, with the population, you see one separate cloud and one big with the others localization. So this one also, the separate is the GBO population, and the same with the genetic population with cluster analysis when key was 2. There again the GBO was totally different than others. Is the green one, and the other trees are pink here. Going further, here we have the genetic environmental adaptation. Here we use different elements to find which part of the environment can explain the genetic variation. So in the climate, it's just the name, but inside we have 5 factors, like mean annual temperature, the daily minimum air temperature of the coldest month, seasonality of precipitation, minimum free precipitation on the warmest quarter, and also the cumulative precipitation on the vegetation season when the plant was growing. So you see the climate explains 8.6% of the genetic variation. Geography is 5, ancestry is also 5, flora is quite interesting because it's 3.6, and inside the flora I would like to say it's a list of the plant which I made on the field close to the trees. So in the all these elements explained 18, close to 19% of the genetic variation. So, adaptive landscape, here I have two plots to show you. First is the Manhattan plot, and here it's all of the loci from the sequencing, but only the orange are interesting for us. They show the connection with the climate. So having those, we can try to estimate, try to say which loci are the most dependent from the climate elements. So, for example, a BO15, it's seasonally of precipitation. So all of this cloud of the loci, cloud of a point, show how many loci are dependent from the precipitation of the season of precipitation, or maybe how the loci are connected with the climate. Here, it's the future of the trees. I also would like to show you two slides about this. Here, this one is more shocked, maybe, because you see two different graphs, and on the first one, here is the RDA1 and RDA2, and they show maybe quite the same, but you see three colors here, like purple, green, and pink. Purple is current, pink and green is the future, the scenario of the climate change, so CCP 370 and 585. And in the graphs, the line inside the purple, you see what is the global environmental issue, environmental scope for the beach right now, and how the environmental issue will be moved in the future because of the changing the climate. So it's look like scared for me. But maybe the trees can live, we don't know. And here, the last map, it's quite similar like here, but to see on the map what's happening. So here is again CCP 370. You see how maybe it's not too good on the presentation, but I see it good. So promise me. It's look like on the lowlands, it's happened the worst in the future, and the environmental issue will be bad for the trees, and the best they should feel on the mountain, like here, when the colors is yellow and a bit blue, maybe. So they then they should feel good and comfortable. But as everyone know, the mountain will finish somewhere, and the trees cannot climbing all the time. So that's all. But I would like to show you some photos. That's me with the brush from the field. This one is my boyfriend with the biggest tree I saw, I think, because in the brave height, the circuit, it was 520. So for me, it was the biggest tree at the beach. And from here, I would like to say thank you for my supervisor who helped me with everything. I promise, with everything. My boyfriend to help me with the field, with collecting the data. Also Sylwia Wierzcholska and Marcin Dziedzic to show me how to collect the leaves from the high trees. And also my university to foundate my research. And also you for the attention. Thanks so much.
Thank you, Olga, for your nice presentation. And the next person will be Mateusz Sydow. He is from the Poznań Institute of Technology, and he will be talking about the utilization of waste generated in the chemical industry for the purposes of sustainable forest management.
So, yeah, thank you. Hello, everybody. My name is Mateusz Sydow, and I represent Łukasiewicz Research Network, Poznań Institute of Technology. I'm not a forester, and I'm not a dendrologist. I am an environmental biotechnologist / chemical engineer, but that's good because I can meet you guys. You are specialists in that case and can help me with my research. Also, big thanks to my co-workers, Kamil and Zbigniew, which are foresters, and their experience is very important for me. But let's start. We will start with the context of the study. So, for most people from Poland, this is probably obvious, but from other people from abroad, it may be quite important. So, Polish forestry traditionally operates on a poor quality land, while this more fertile land is mostly intended for agricultural purposes. And in years between 2020 and 2022, about 2,000 hectares of agricultural land and wasteland were afforested in Poland. However, about 150,000 hectares of fallow land in Poland are still available for afforestation. However, there are many aspects that have to be covered when speaking about deforestation. So, as you probably know, deforestation on post-agricultural land can be somehow limited by the, for example, loss of essential elements or worse quality of the land. And pre-fertilization of these lands may improve soil properties and increase the success of forest crops. However, there are economic, environmental, and geopolitical limitations and reasons that are reducing currently availability of the conventional fertilizers. On the other hand, there is a tendency to increase the acreage of the protected areas in Poland, protected forest areas in Poland, so the potential area for timber harvesting will be also limited in the future. Therefore, introduction of alternative fertilizers based on waste or byproducts may help mitigate this problem. So here comes our idea for alternative fertilizer, phosphogypsum. Phosphogypsum is basically a calcium sulfonate with water, with phosphoric acid and many different elements that is produced during the production of the phosphoric acid. So during the production of 1 ton of phosphoric acid, about 5 to 6 tons of phosphogypsum are generated. Currently, about 100 million tons per year are producing worldwide of the phosphogypsum. Unfortunately, only about 3 to 5% of this phosphogypsum of the waste is generally used somehow, mostly in the construction, material construction, for example, in construction materials mostly. However, so about 95% of the phosphogypsum is still on the landfills and cannot be used. So chemical engineers and the producers are looking for new ways of utilizing that waste. So we wanted to find out if it can be used as an alternative fertilizer. However, it's not that simple because, as I said, phosphogypsum can contain phosphorus, can contain calcium, which are very essential elements that can help increase the crop's growth. However, it also contains heavy metals. And it depends, the contamination, or I would say the content of the heavy metals, depends on the mining site or the mining countryside of the raw material. So, for example, there are different contents of the heavy metals in phosphogypsum originating from Morocco phosphorites, Tunis phosphorites, and Kola apatite. But there is a question: is this dangerous? Is that quantities are dangerous for the potential forest management people, or for the trees by themselves? So the aim of the study was to assess the potential of Scots pine seedlings for the short-term absorption of selected heavy metals from the soil fertilized with the waste material generated during the production of phosphoric acid, which means phosphogypsum. So we started with the stage one. In the first step, we tried to develop this alternative fertilizer using two different components. So the first one was, of course, the phosphogypsum, but the second one was wood ash. And both of those elements, those components originated from one of the biggest production entities producing phosphoric acid in Poland. We added the wood ash to the phosphogypsum to make it more, make the pH of the fertilizer more neutral because of the contamination or the content of the phosphoric acid in the phosphogypsum. And wood ash is mostly alkaline. So in the first stage, which turned out was not very successful, we just conducted preliminary study which lasted only 5 months. We used forest soil and Scots pine seedlings, 1-year Scots pine seedlings from Trzebież Forest District in Poland. And the research was carried out only in 2 variants. Well, the control, which was not fertilized with the phosphogypsum, and the second variant was fertilized with the phosphogypsum at the dosage of 5 tons per hectare. We used it in experiment was used in pots with dimensions of about 18 per 18 per 20 centimeters. And the exposure for the fertilizer lasted only 5 months. Unfortunately, we were waiting for the supply of the phosphogypsum from the producer. Unfortunately, we did not get it at the right time, so we started the experiment on May, not on April, not before the spring growth season. So the results were not very conclusive. However, we measured the content of heavy metals, mostly cadmium, lead, nickel, and copper, in wood tissues and soil, to find out how the, let's say, absorption of the heavy metals from the soil and a possible migration of these heavy metals through the wood tissues developed during the, well, I would say, very short experiment. And well, the results were in general not very conclusive. We found out, for example, in the lead context, that the contamination of lead was higher in the soil where the soil fertilizer was deposits. However, and also we find out that the content of the lead in wood tissues was mostly visible in the root tissues. However, the differences between the control and the variant with the dosage of 5 tons per hectare were not really significant. And this is one of the examples here for the copper, where the results were totally not conclusive for us. So we decided to start again and try not to make any mistakes again. We had 4 main conclusions from that first stage. So the number of seedlings per research variant should be greater than 4. Moreover, more research variants should be tested. then the test start month and its duration may be crucial for metal absorption. And the last, the lowest layer of soil, which was not tested, can be really important, as the highest content of heavy metals could potentially be accumulated, let's say, at the lowest layer of the soil. And at stage 2, I call it short-term. Well, it's a long-term, it depends how you call it, but because from the tree point of view, it's a short-term, but from our perspective, it may be a long-term test, which lasts in general 30 months. We collected soil from near Rogalinek, which is very close to Kórnik. We used also 1-year-old Scots pine seedlings from the Babki Forest District. And this time research, this is an ongoing research, we are conducting that in 8 variants. So control and 3 different dosages of the fertilizers. We also have bigger pots with more of the soil, and we divided it for 2 parts. So first half of the containers are exposed for 18 months, which is about 2 vegetation seasons, while the other half of the containers will be exposed for 30 months, so about 3 vegetation seasons. The experiment starts in April 2023, so it's still ongoing. And we will perform analysis of the content of the heavy metals, the same heavy metals as before, in wood tissues and soil, all of the layers of soil, and analyze the growth rate of the seedlings. So here is a static time lapse of the experiment from, let's say, April to February 2024. As you can see, we measured before cultivation, we measured the seedlings, and you can see that the growth of the seedlings is quite good. We do not observe any acute toxicity of the fertilizer, as we did not observe it in the first stage of the experiment. So that's kind of promising. Of course, we will see how it goes. We will finish the first part of the experiment on probably on September or October 2024. So it's quite near. Only 2 of the seedlings died in the initial phase of the experiment, probably not because of the fertilizer or any other, let's say, crucial aspects of the experiment. Probably they were weaker than others. And here's the current stage. The photo is from beginning of May in 2024. Yeah, so we can see that the spring growth is quite visible, and the seedlings seems to be okay. Unfortunately, we have some problems with the forest pests, with the pine web spinning sowfly and pine shoot moth. Of course, we are trying to reduce their quantity and their presence mostly in the mechanical ways, because we do not want to introduce any chemical intervention right now, which may, influence the results, because some of the, let's say, some of the chemical components may influence the growth of the trees and stuff like that. And they can also contain, for example, copper. So we are trying not to use any chemical substances. Yeah, and that's probably all. Maybe not all at all, but because I'm looking for a cooperation with you guys, especially because, we are looking for, we are, studying the influence of the fertilizer on the Scots pine seedlings, but it will be probably quite nice and quite important from the scientific point of view to find out the influence on the microbial, soil microbiota, probably the structure of the community, maybe fungal community and stuff like that, and it could possibly increase the scientific level of the research. So I'm looking for a cooperation with you guys. If you can help me, then it will be great. Please contact me. Here is a mail. And thank you for your attention. Thanks.
Thank you, Mateusz, for your very interesting presentation. And now,
No, to już na razie tak.
the last presentation will be given by Katarzyna Ewa Niedźwiecka from University of Warsaw. Yeah.
Mogę klimę na prelegenta?
Słucham?
Klimę na prelegenta mogę?
To za chwilę, bo to muszę jej zgłosić indziej. Ale proszę.
Okay. Hello. Who tried to do popularization of science or wants to do this? No one, maybe you will try later. And who of you tried to take part in some popularization event and could not do it because of lack of time or limit of participants? Okay, one person, me too, 2 people. Today I will tell you about way of popularization with no limit of participants, no limit of time. You can learn whenever you want. And it's geocaching. Geocaching is a game for GPS users. It's a kind of treasure hunting. This is a typical view of a geocache. It's a moisture-protected box with some various content hidden somewhere at certain coordinates. And in the internet, it's written where it is, how to find it, and what is interesting around, because the aim of geocaching is to show interesting places. A brief history of geocaching. Geocaching started when a global positioning system started being available for civilians. And one day later, first geocache was created. Then web pages that gather various geocaches were created. This one exists till now. And we didn't wait a lot for geocaches in Poland. And 5 years later, the biggest non-commercial web page for geocaches were created, and it was in Polish. This one is commercial, and it's American and worldwide. So, how to use geocaching for popularization of science? Because you probably want to show your knowledge, your places to people. Every geocache has a description. You can put, it's an example of existing description of existing geocache. I translated it. It's close to Częstochowa. And the author created his geocache, put it in the forest, and made a description. And the participants go to these coordinates, degrees and minutes with 3 ciphers after comma. It's enough, the accuracy is maybe 1 or 2 meters, and it's more than GPS in your phone, usually, because it has some limitations. And the participants go there, finds the geocache, logs it, and maybe he or she reads the description or not. but you want to make sure that the participants take this knowledge, and you can also put your educational material inside the geocache. So the participants go to the forests, go to the coordinates, and see. It's a typical, one of the quite typical maskings of geocaches. It's a fake bird house, sorry. And you can open it. And may I ask my volunteers here? Yes, and you can tell your participants that the final box, when they can find the geocache and log it, is a bit enigmatic. So it's 52 degrees and 14 point pine, oak, spruce, and and it's north, and east is birch, beech, and maple. Normally you use ciphers from it, but here we don't have ciphers. We have where the place is, and we see that the place is, thank you so much, the place is behind Institute Dendrology of Polish Academy of Science, and we come to this place and look for the geocache, and we are lucky we found it. Take one and give it to next person. You can also use your take-home message on the certificates for the finders. People like certificates. And sometimes your knowledge is already there. Sometimes there are educational boards. And you want to show, but you are not sure whether the participant reads and learns, or he or she just picks the geocache and runs to pick the next one. But you can hide your geocache in a place when you cipher the coordinates. So the participant has to pick some information from the education board. So let's say you have already chosen the place that you want to show, maybe a lake, maybe a particular old tree, maybe some historical point in your forest, maybe certain type of ecosystem, and you want to prepare your geocache, and you worry about the environment. So think well how to create and hide your geocache. Such content is forbidden. Don't put food because an animal smells and eats and dies and destroys your cache. Make sure that you don't destroy animals' home, or that an animal can't use your geocache as a house. This hole should be like this. it shouldn't be empty so that a bird comes in and the geocacher disturbs this bird in living. Don't put nails in the in living trees. Don't make hollows bigger, and remember that the finder should also respect the environment. So describe well where your geocache is, so that the finder doesn't dig up the soil, doesn't enter all the hollows that he or she finds. And you can also organize or take part in a meeting where we collect trash in the forest. It's also a meeting that this meeting was last September, and we collected trash on this way, and then the authorities went here by car and collected the trash that we collected from the forest and put onto the road. More people attended. We were about 20, and it was a very nice meeting. And I also want to tell you something about geocaching in national parks. First of all, every geocache in the national park has to have acceptance of the head of the national park, and it's quite easy to get. It doesn't require a lot of materials and methods. Usually you can take paper from the secretariat, and people are helpful. This is my application for a geocache, for acceptance of geocache in Biebrzański National Park. Some national parks allow virtual geocaches without asking for permission. This turquoise color means virtual caches. There you have to take a photo or type a password instead of finding a physical box. And you see there is quite a lot of geocaches here. Some of them indicate historical points, some of them are strictly connected to nature, and probably all of them are interesting. And the best thing that the national park can do is cooperation with geocachers. In this case, National Park of Góry Stołowe, where we are going, applies this. It has geocaching in their strategy. They employ volunteers to create geocaches, and it has many advantages. And the national park has full control where the geocaches are, and sometimes you can get some funding so that your geocache is really great, not just a box, but in a supermarket. And at the end, I'm inviting you for geocaching. There are a lot of really nice geocaches here in Kórnik. The closest one is, we are here, and the closest one is on the bridge over there, 150 meters from here. And then in National Park of Góry Stołowe, I'm staying there for a few days. Yes, and I have to acknowledge many people who helped me with some aspects of preparing of presentation and abstract and the poster, and geocachers who went for geocaching with me, and the authors of the caches that I showed you as examples. and of course my lab. These are sources of graphics and other things, and thank you for your attention.
Thank you, Katarzyna, for your very nice and
full of gadgets and fireworks.
Presentation. And now we finish this part, and I would like to thank every speakers from sharing the results, preparing the very professional presentations, and for the time they respect. So now we will start a discussion, and we have about 15 minutes for it. So please ask the question the speakers. Maybe to speaker one or.
I have a question to the speaker number one. First, Mr. Guidos, you, with your research, you exclude the hypothesis of the mycorrhizae on the monodominance of Ocumea cliniana populations in Gabon. What can you suggest or put as a second hypothesis as you exclude this one?
Yeah, thank you for the questions. Yeah, so as I said, main hypothesis about the monodominance would be about the root grafts that are happening, and then so those merged root systems would take some more spaces in the soil, not giving it for other species, so it would be a reason. I also wanted to ask a question at the last session to the person who was online about it, because she also worked about monodominance, but it wasn't possible to ask her some questions. She worked on litter chemical composition, so it would be a reason as well. For example, we know that for Okume, it creates its own litter in the forests, and that it might be the reason why it is the only one that can grow on it in the beginning. And yeah, another reason could be light competition and the fact that they grow into dense stands, but it would be linked to the two other regions that I've cited already. So yeah, that's what I think.
Maybe next questions to this speaker, or maybe for another one.
Thank you again. I have another question addressed to Mrs. Olga. With your PCA, you captured 8% of variability from genetic. That was explained the adaptation to warming of the species you studied. And the 8% of variability was related to axes, the first three axes. A huge part of the variability was not explained, and I want to know as it can be explained on the phenotypical plasticity of the species to environmental adaptation. What can you give us as an information concerning the plasticity adaptation of that species in the variability in the environment?
I don't know. I'm sorry. I think I can't answer the question, or maybe I don't understand you too much. Can you explain it in a bit short?
If I can be more concise, your PCR explained 8% of variability that is captured by genetic of the species. Then a huge part of variability was not explained by your analysis. My question is it related to the capacity of the species to adapt to the environment, related to what can I say, the capacity of the species, if you change the environment, it adapts to the new environment. If, for example, you you change the site where you studied and you do the same study, you think that you will have 8% which is explained by genetic variability, or it can be higher than that you captured on your study site?
So I think with these samples, what I can have explained is quite big, because for this all I have only 84, so it's not a lot. But climate, I think it's also quite big for the explaining. But also we have to remember the forests are also the management, because the foresters. So big part of the genetic variation can be different because the different seeds using on this place. I hope I answer a bit. Maybe.
Thank you. Oh, another question.
So I have also a question for our European beach presenter, because you mentioned that you had help with methodology to collect leaves from trees, and I believe that some of the old trees have leaves very high above the ground. So can you, in short, explain how did you collect them? Did you use ladder or something like that?
We didn't use ladder because my friend is the arborist. He showed me how to collect leaves without climbing on the trees. So for that I used a bucket with long rope inside. I think it was like 8 meters, with the big, not big, with the little bag, heavy bag. And then when we found the nice tree for the correct the data, we make the triangle across our hands, and using the bag, did something like the swing like this and throw us far away. And sometimes we catch the branch, sometimes not. So we spent the longest it was, I think, half an hour try to catch the branch, but finally we have everything, so it's working.
I have first a question to Hanna. So you said that sycamore maple has the more genes that are related to high metabolism rate in their seeds, right? So are there any relations of those genes and, for example, tree health or growth rate?
First of all, we were studied proteins, not genes. We identified the genes and the proteins, And as I mentioned at the end of my presentation, our results complement the results of Eva Kalemba's research. So maybe after the discussion, I will show you her her article. Yeah. I think her article explained it very well.
Now, Mateusz, please. You said that stuff you collect for the fertilizer, for potential fertilizer, contains heavy metals. So, are you not worried that they may end up in the groundwater?
Yes, of course. That's one of the risks. Yeah. So, we also collect some of the water that is going through the soil, and we will also finding out how much of the heavy metals content can be in that, I would say, outflows from the containers, and we will see if that could be potentially dangerous for the environment. Because of course, in the soil environment, it's natural that these heavy metals will migrate in time. Horizontally or vertically, we don't know, but we know that it's a very important topic, and we will find out it, hopefully.
Now, another one for you, it's about the legality of all of it. Now, I'm not a forester neither or a lawyer, but when I worked with some foresters in Estonia, they told me that in the entirety of the European Union, it is illegal to treat your forest with something that is labeled fertilizer.
Well, in Poland, it's illegal to use it as a waste, but the producer is working on it just to change it not as a waste, but just to treat it as a byproduct. And when you have a byproduct, there are many, much more ways to utilize that type of material, also in the forestry. So we are here to find out if it's really dangerous, and we are doing that, let's say, hoping that the producer finally can utilize it as a byproduct and make it formally in law as a byproduct. But right now, it's just a waste. It's a waste material that can potentially be harmful. We don't know. We want to find out right now. Because there are heavy metals in that material, but we don't know if this content of heavy metals can be potentially dangerous or not. Maybe it's not that really severe.
So maybe another question to the another presenters. Oh.
It's a question to Hania, because you said that these three species are the good models for the molecular analysis. So I think it's sometimes difficult because they do not have some annotations and you don't know the function. It's better used to Arabidopsis for sure, or Poplar. And how you found the function for the classification? Just from these trees, or just looking for the homologs, orthologs?
We were looking for orthologs according to KEGG databases. So we annotated them according to Arabidopsis and other models.
Eva Kalemba.
I have also a question to Hania Fuchs. There are plenty of thyroidoxin in the plant kingdom, plenty types. So which type of the thyroidoxin did you use in your experiment? Was it a specific one? And if it was a specific one, please explain what was the criterion to choose this one.
We used thyroidoxin H1. And as I mentioned at the beginning of my presentation, molecular biology might be tricky. as Paweł said, we don't have many reference genomes, so we had to use a thyrodoxin from peas. But the active site is highly conservative, so we thought that this method might actually work on our material, and it did. Thyrodoxin H1 was chose because of its present not only in the cytoplasm but also in mitochondria, which are our scope of interest, and that was the main reason.
Any questions? Yeah.
Thank you very much. Another question maybe for Hannah. Thank you very much for the very nice presentation. I was just wondering, when you mentioned at the beginning the comparison across the different storage behaviors, maybe I miss it. At which moisture, did you use the same moisture conditions for all the species, the 3 seeds?
We used, first of all, we used mature seeds, that they were fresh, and they were stored according to the ISTA rules. Yeah.
And one question for Katarzyna. Is your game popular in Poland, especially in schools and in older people? Because I have never heard about them, so maybe you tell something about this.
Well, the game is, I don't know whether it's popular. The problem with popularity is that we don't have funding for advertisements, and usually people know about it from other people, but there are also some actions connected to, I don't know, some local festivals or something that popularize it. You asked about popularity in schools. Yes, in Warsaw, in Saska Kępa, there was a teacher who played geocaching and created many geocaches in Saska Kępa showing certain buildings, and it's quite a lot. And these geocaches are reactivated by next generations of geocachers, but the description is still there. I don't know how to define popularity. If you define it as a % of people who know about it, it's maybe not so many, because there is also a problem with hiding geocaches that your foreign people shouldn't find and destroy it. So when you are looking for a geocache, first look around whether no one is looking at you. And it's especially a problem in the cities. And we also have meetings, so you can meet, on one meeting you can meet more than 100 people in the some days. Maybe I should stop talking about geocaching now.
So who heard about this game here? Good statistic. Okay, 20%.
Thank you for your answer. And Dominique?
And I have a question whether everyone had opportunity to pick a take-home message from the red bag, including the guys with camera and so on.
thank you. I have one question to Michał about your study, because you studied fungal communities in, for example, in spruce and birch. Birch is much simpler, I think, in terms of longevity of the leaves. However, what about the spruce? Because the needles can have several years. How old were the needles you used for your research? And what about the evolution of communities in the needles? Because I suppose they can be different in different years.
Well, you're right in the sense that the spruce fungal communities are quite complicated. My needles were 1 year old, and my trees, I must say, were also really young. They were planted in 2020, so they're by now about 7 years old, I think. So the evolution of fungal communities in those several-year-old needles is not very well quantified, I must say. So the spruce starts out with less fungi, different fungi in their needles, and the colonization rate is at first lower, but as the years go by, the colonization rates become higher and the colonies become more densely packed, and at some point they reach a certain critical threshold, you may say, which contributes to the needle senescence and can be partially responsible for the variation in the longevity of the needles. So in some places you may see that the needles fall off at 4 years, in some places 5 or 6.
Thank you very much. We will finish now, and I would like to thank you for the presentation, fruitful discussion, and I invite you for the lunch now, and enjoy the stay in Kórnik and in Góry Stołowe in Poland.
I'm from Institute of Dendrology. I have the pleasure of being your chairperson of this session. As you must have something interesting for herself or himself. Now we can start with the presentation of Lukas Petrulitis, if I can pronounce it well. Lukas is from Nature Research Center in Vilnius and will talk about less productive but successful case of Kórnik sericea invasion in Lithuania. So please start.
Thank you very much. So I hope you won't fall asleep after this heavy lunch. And for an introduction, I would like to tell some facts about alien flora of Lithuania. So alien woody species comprises only 12% of all alien flora in Lithuania. That comprises only 83 alien woody species out of 700 alien plant species recorded in Lithuania. And just few of these woody species occur in Lithuanian forests, such as Amelancher spicata, Cititus coparius, or Robinia pseudoacacia. These species alter soil properties by nitrogen fixing and changes the succession of these forests. But nowadays, newly emerging alien species occur in Lithuania, for example, as Celastrus orbiculatus is literally strangling the trees by forcing water uptake, and the tree just dies off. And nowadays, Kórnik sericea also has started to penetrate into alluvial forests, and I would like to talk about it more in detail. So, in Lithuania, 7 species of the genus Kórnik have been identified belonging to 3 subgenera, and 3 alien species originate from North America. This is Kórnik amomum, Kórnik alternifolia, and Kórnik sericea. Here you can see Kórnik alternifolia and Kórnik amomum. And actually, Kórnik alternifolia is very rare alien species in Europe. And Kórnik amomum as an alien species was recorded only once by us in Lithuania. It was a new record for the whole Europe. And Kórnik alternifolia, I know that this is spreading even in Kórnik arboretum. There is a nice paper of Marcin that he published 7-8 years ago, I think. Other species that occur in Lithuania, such as Kórnik alba, came from Eurasia. And Kórnik mas and Kórnik sanguinea subspecies australis came from southeastern part of Europe, and they are treated as aliens in Lithuania. So only one species, subspecies, Kórnik sanguinea subspecies sanguinea, is native to Lithuania. So our objective was to evaluate the biological and ecological traits of the genus Kórnik in the Baltic region, and to characterize traits that define their invasiveness. And to study, we wanted to compare reproductive characteristics of Kórnik found in the country. So we studied flowering and fruiting traits. Also, we wanted to evaluate the composition of the soil seed bank and density of seedlings, and to see their recruitment. Also, we compared the annual ring increment of studied Kórnik taxa, and we assessed the communities with Kórnik sericea in Lithuania. So there is little data on threat posed by plants of the genus Kórnik, and Kórnik sericea naturally occurs in North America. It covers very wide part of the continent. And even though the species is often included into the list of alien plant species in several countries, there is no data justifying the invasiveness of the species. So even in the native range, sometimes the species is treated as invasive, even though it should be treated as only as expansive, not as invasive, as it occurs in its native range. So for this study, we the research was carried out throughout Lithuania in order to evaluate the traits that determine the distribution and invasiveness of these alien Cornus taxa. So for the soil seed bank study and seedling recruitment, we selected 5 sites of each taxon of Cornus alba, Cornus sericea, Cornus sanguinea subspecies australis, and native one, Cornus sanguinea subspecies sanguinea. So we collected 400 soil samples that were divided into 2 layers, upper layer from 0 to 5 centimeters and lower layer from 5 to 10 centimeters. And in total, we got 800 of soil subsamples, and we extracted seeds from these subsamples and compared them between species. And also, we assessed the viability of seeds extracted from these samples. In the same sites, we carried out seedling recruitment study. We assessed 400 disturbed and 400 control plots. Also, we compared flowering and fruiting of selected Cornus taxa. We collected 30 inflorescences and afterwards 30 infructances, and we counted flowers and fruits in each site. And also we compared radial increments, also at 5 sites of each taxon. In total, we collected 600 wood samples. And also we assessed communities with Cornus sericea at 30 sites. So the first results of this reproductive output shows that even it seems that Cornus sericea has significantly more flowers and fruits than Cornus alba, its congeneric species. The potential yield of this species is actually lower. And this indicates that even the higher number of flowers does not lead to a higher number of ripening fruits. And the studied Cornus alba populations had a potential yield that varied from 18 to 41%, whereas at sites of Cornus sericea, the potential yield was less variable, and it varied from 24 to only 26%. So we also evaluated the composition of the soil seed bank and density of seedlings, considering the statements by Margherita Gioria on differences of the seed bank structure between native and alien woody species. And we hypothesized that alien Cornus taxa forms larger seed banks than the native Cornus sanguinea subspecies sanguinea. So examination of soil samples revealed that the seed bank of Cornus taxa was statistically significant between sites and within sites, and the same tendencies were observed in the upper and in the lower soil layer. Here we can see that Cornus sericea had very low densities of seeds in the seed bank, both in the upper soil layer and in the lower soil layer. And the largest densities of seeds in the seed bank were reported from Cornus sanguinea subspecies australis sites, which is an alien subspecies in Lithuania. The density of viable seeds in the seed bank of Cornus sericea sites was also significantly lower. But here it seems that the species has a huge number of viable seeds, but as I showed you before, the actual number of seeds in the seed bank is very low. So this doesn't reflect the whole picture of the species, as it has not very dense seed bank, but these low numbers of seeds in the seed bank are very much viable. And after evaluating the disturbed and control plots, it was found that in all individual Cornus populations, there were no significant differences between disturbed and control plots, except for Cornus sanguinea subspecies australis sites. And here you can see a seedling proliferation of Cornus sericea in forest glade after removal the mature individuals. And our experiment was quite based on what we saw in this site, because we thought that if this disturbance caused such huge seedling recruitment, so it might be useful to carry out an experiment with soil disturbance, but here we can see that Cornus sericea has very low rates of seedling recruitment, both in disturbed and undisturbed plots. The cumulative annual ring width patterns of the alien Cornus alba and Cornus sericea was similar. Here you can see Cornus sericea in purple and Cornus alba in red, but and the mean annual ring width patterns were similar, and they were not significantly different, but results of two-way nested ANOVA shows that effect of the taxon and the year was significant, as well as their interaction. And after evaluating the growth rate of individual taxa, it was found that the cumulative annual growth of Cornus alba and Cornus sericea species was similar both in different years and also by the general trend. And also we observed that the alien subspecies Cornus sanguinea subspecies australis grows much faster than the native one Cornus sanguinea subspecies sanguinea. And this shows that the alien taxa of the genus Cornus is more invasive with comparison with the native one, as it grows much faster, and this faster growth can be one of the characteristics that determine their invasiveness of these species. Also, a statistically significant negative correlation was found after the assessment of communities with Cornus sericea. We found communities where only 2 or 4 species of plants were present in these communities, and this negative correlation between Cornus sericea cover and number of herbaceous plants can be explained by the shady conditions that occur under stands of Cornus sericea. And the evaluation of these communities show that most of the species that are present with Cornus sericea belong to Alinopadion association, and nowadays Cornus sericea penetrates into alluvial forests and affects the vegetation of these forests by suppressing the native vegetation. So even though there are still many questions that require a proper answer, and even it seems that it's less productive than its congeners and the soil seed bank is not the densest, but maybe fast growth and vegetative spread by stolons compensates this the lack of in these traits. And in some cases, even the seed where few seeds were present in the seed bank, the viability of the seeds was very high, and maybe this also the fact that leads to its successful invasion. And also propagal pressure and spread by other vectors was not assessed. And this is the map of Cornus sericea in Lithuania. We had a project of mapping all invasive species in Lithuania, and we can see that there are no empty grid cell of the species. Of course, the species was found in various habitats, not only in forest habitats, but nowadays we are seeing that as its propagal pressure is very increased, it penetrates into alluvial forests and grows there very well because of the huge amount of nitrogen and humidity. And future insights into invasion of ecology of the species, it was shown that in the native range the species has very high evapotranspiration rates, and this also indicates that the species can alter hydrological regime in specific habitats. So maybe there should be a study carried out in changes of the species can cause in the hydrological regime in the same alluvial forests. And also in the native range there was a study carried out about 2 types of shoots that this species produce. In open habitats, the species grows erect and does not tend to be invasive one, but under closed canopy, the species creeps along the soil surface until it reaches open space, and then it starts to grow very fast. So I think these studies should be carried out also in Europe, because now there is no justified data about the invasion status of this species. Thank you.
Yes. Thank you very much. I think we will put aside and for later, save it for later our questions. So now, please proceed with the next speaker, who will be Katarzyna Kondrat from Adam Mickiewicz University, and she will talk about drivers on intraspecific variation and in fecundity in rowan. It means Sorbus aucuparia. So.
Hello everyone, my name is Kasia and I work in Forest Biology Center, and today I will present you my research that was my master thesis and that I published a few months ago. So tree fecundity, that can be defined as a number of seeds or fruits produced per tree, is important for really numerous reasons. And I'm only gonna name a few that I think are the most important ones. So first of all, it's important for forest regeneration, as it is a key factor that determines the reproductive success and overall population dynamics, and can also help in predicting the distribution and abundance of tree population. Also, seeds constitute the base of food webs and can influence ecosystems through trophic cascades. And in the end, it's also important for ecosystem services, such as, for example, soil nutrient cycling. And although it is important, in my opinion, it is an area of tree ecology that is severely understudied. And that is mostly because it is challenging to measure seed production. And what adds to the challenge is the fact that very often trees don't produce seeds every year, and that change can span several orders of magnitude. And the factors that influence tree fecundity can basically be divided into two groups. So the first group are intrinsic factors, and those are the ones that are connected to the tree itself. So that's either tree size, so DBH or height, and also leaf nutrient availability. And the other group are extrinsic factors, so the ones that are connected to the environment, and that could be light availability, so nutrient availability, and also elevation that is important in mountainous regions. So in my research, I had two main objectives. So first of all, I wanted to find the best size correlate of fecundity, because obviously it's a no-brainer that the bigger the tree is, the more seeds it will produce. That's usually the case. But I wanted to see if it's DBH or height that better correlates with fecundity. And having found that correlate, I wanted to see how other factors influence fecundity on per-size basis. So per this best size correlate that I found. And my study species is rowan, Sorbus aucuparia. It's an insect-pollinated tree species. It produces fleshy fruits, which makes it actually easier to count them and therefore estimate fecundity. And fruit production is intermittent and highly synchronized between individuals. So that means that rowan is a mast-seeding species. And so they're dispersed mostly by mammals and birds. And the study was conducted in Babia Góra National Park in southern Poland. It's in mountains. And here is the map of the whole park. And the red rectangle is the study plot that is situated in subalpine spruce forest. And then here we have the map of the plot. And each red dot is an individual tree that we took into consideration. And in total, we had 167 trees. And so for the data collection, first of all, we've conducted fruit counts that started back in 2000. Obviously, I joined like a few years ago because in 2000 I was like in preschool, I think. But they were done using binoculars. And here's the fruit production data. So on x-axis, we have year. On y-axis, number of fruits per tree. And each individual line is an individual tree. So as I mentioned, you can see here mast-seeding pattern, which means that there are some years where there's virtually no seed crop, And then you have bumper years, where there's a lot of seeds and all the trees pretty much produce them in synchrony. There are maybe some outliers. So the next step was to collect soil samples. So 5 samples were taken from around the tree, and we measured the concentration of nutrients that we believed could have the biggest impact of fecundity. So that was carbon, nitrogen, phosphorus, calcium, and also other cations. We've done something similar for leaf nutrient content. Here we collected around 20 healthy leaves. And again, we measured the content of elements that we believed could contribute the most to the fecundity. Then we measure light availability by taking photographs from above the tree crown. And having that photos allowed us to measure percentage of total light available to the tree crown. And in the end, we also measure neighborhood crowding, but we separated it for conspecifics, so for the rowan trees, and for heterospecifics, so for the rowan and also trees of other species that was mostly Norway spruce. And the formula looks somewhat intimidating, but basically what it takes into account is our central tree, its DBH, and then the distance between the tree and neighboring trees, and also the DBH of neighboring trees. And long story short, the bigger is the value of this index, the bigger is the competition. So in order to determine the best size correlate, I've fit a model where as a predictor, I've put either DBH or tree height, and as a response variable, fecundity measured as a number of fruits per tree. And it turns out that DBH actually was a better size predictor than height, as seen by the AC value. And here on graph A, on x-axis, we have DBH. On Y, the fecundity, so the number of fruits per tree. And each dot is an individual rowan tree, so you can see also that the bigger is the tree, the more fruits it produces. And that's pretty much the same story for height, but nevertheless, DBH was a better predictor. And so having found that best predictor, I wanted to see how other factors influence fecundity. And I've fit a pretty robust model where I've put light availability, so nutrient content, also leaf nutrient content, elevation, neighborhood crowding index for conspecific, and also for heterospecifics. And as a response, I fit fecundity, but this time it was standardized by DBH. And it actually turns out that only 2 of those predictors had an impact on fecundity. So that was light availability and crowding, but only from rowan trees. So as you can see here, on A-axis, we have light availability measured in the percentage of light available to the tree crown. On Y, we have number of fruits per tree, but standardized by DBH. And also, all of those values, they were scaled in order to be able to compare them. So that's why the value only goes to 2. And here again, each dot is an individual tree. So here we can see a very strong positive effect. And this strong impact of light of fecundity can be attributed to the fact that light just increases photosynthetic capacity and availability of soluble sugars that is important for fruit production, essentially. And on B panel, on x-axis, we have the crowding index. On Y, again, standardized fecundity. And here we can see a negative effect. And that can, I think, be mostly likely to be attributed to the competition for pollinators between the trees. And so to conclude, DBH was a better size predictor than tree height, and the individual tree fecundity was positively correlated with light availability, but negatively correlated with crowding from other rowan trees. And so all those results can be used as a recommendations for managing high light availability and also low conspecific crowding. And again, it's also a contribution to better understanding seed production ecology. And I would like to thank everyone who participated, especially my supervisors, Magda Żywiec and Michał Bogdziewicz. Okay.
3 minutes, so I would like to
Sure.
ask you one question.
Yeah.
Because you mentioned here that rowan doesn't have fruits each year, and I noticed that in 2008 and 2014 they were like null years.
Yeah.
And what do you think? how can you speculate about the impact on animals which feed on the fruit? Because this is dramatic loss
true.
for them. Yeah.
So I haven't done a research on the seed dispersers. I think that it definitely can have an impact, but also we have to remember that it's just a one study plot where it took place in the upper layer of the forest, and then in the lower layer of the forest, I think there are also other tree species that can potentially be used as a food for those animals. But also the fact that there are the years when there is no seed production is actually good for the rowan, because there's this apple fruit moth that is a seed predator. So during the years when there's no seed production, it essentially starves out the whole population. And then the next year, when there's no population of those seed predators, the rowan just does better because it can produce more fruits.
Okay, so it's like a clearance of,
Yeah,
the
exactly.
enemy. Okay, thank you very much for this, because it looked really, really dramatic, for other organisms who are related to the rowan. So thank you very much. And now I think we can skip to the next speaker, who will be Pulak Majra from the Institute of Dendrology. And Pulak will talk and present us his topic is that is effects of Pinus sylvestris, or geographical origin, on the community and co-occurrence of fungal and bacterial endophytes in a common garden experiment. So you're welcome.
Good afternoon to everybody. I am Dr. Pulak Mitra, working in the Institute of Dendrology as a postdoctoral researcher. So today I'm going to talk about the effect of Pinus sylvestris geographical origin on the community and co-occurrence of fungal and bacterial endophyte in a common garden experiment. So here we all are plant scientists, so we may know more or less about the endophyte, I think. So an endophyte is any microbes that typically live inside the plant without causing any disease, and have an important role in plant growth and fitness. Some endophyte may be recruited from the soil, but similarly benefit to the plants. Now I'm going to talk about the relationship of endophytes and plants under climate change. And climate change, previous study mentioned that the climate change influence endophytic assembly and growth of an invasive plant in response to climate factor. For example, here in previous study, we can look that the drought and temperature influence root and leaf endophytic diversity and ultimately influence the biomass of the plant. Previous study also demonstrated that the genotype variation lead to the distinct ectomycorrhizal composition associated with some plant trait, like some structural compound, for example, carbon, nitrogen, phosphorus, and nutrient mineralization. But still we don't know how ecotypic variation of plant influence root endophytic community assembly. So our aim of our study is to examine the influence of tree origin on the community and co-occurrence of root endophytic fungi and bacteria in translocated Scotch pine in Poland across different growing seasons. So we have some hypotheses. The structure of root endophytic communities in Pinus sylvestris will vary in tree ecotype because of the variation of root biochemical phenotypes. The association of root endophytic community in Pinus sylvestris of different origin is season dependent because season have big influence on the plant metabolic activity. And co-occurrence of endophytic fungi and bacteria is influenced by geographical origin of pine, as their interaction may facilitate plant growth and promotion. So we have a harvested root from 60 individual tree of Pinus sylvestris, who is representing 6 geographical origin. And we have surface sterilized the root, and we have extracted the DNA. And fungal and bacterial community are analyzed using Illumina MySeq sequencing by amplifying the 16S and I2S region. We have also collected some data from the climatic data from the Chelsea database for each geographical origin. And we have also measured some root biochemical parameters, for example, esters, TNC, carbon, and nitrogen. Now I move to the result section. And we have found some significant difference in some root biochemical parameters. For example, especially in root esters and total non-structural carbohydrate. And here we can look that in PPN region in spring have the highest root esters and total non-structural carbohydrate. Compared to the BK origin. But this difference is not prominent in autumn, only in spring. However, we didn't find any significant difference in root carbon and root nitrogen, regardless of the season. This may be because you can have a look the higher peak in spring, because in spring this PPN may produce store more photosynthetic carbon because this region, origin of this geographical region is mostly precipitated and cooler region. So we guess that they store more photosynthetic carbon in this geographical origin. So now I am going to talk about the alpha diversity indices. And we found that tree origin didn't significantly affect the OTU richness or the Shannon diversity index for both fungal and bacterial endophytes. But we found significant influence of season on the richness of bacterial OTU and Shannon diversity index of fungal OTU. We didn't find so much difference in the alpha diversity indices because maybe species pool in a common garden experiment is remain constant rather than in a natural habitat. Now I am moving to the relative abundance of endophytic fungi. And heat map analysis demonstrated that the relative abundance of some specific endophytic fungal OTU within pinus silvestris exhibited biases based on the seasonal change and the geographical origin. I mean, some geographical origin and in some season have a difference in abundance of mostly abundant fungal OTU. For example, here, OTU 39 and OTU 55 is abundant in BK in spring, and OTU 59, OTU 54, and OTU 70 is more abundant in PPN region in autumn. This is maybe because of the combination of host genetic characteristics from the different origin and the climatic difference in different season. Similarly, the bacterial relative abundance of bacterial OTU shows the similar trend like the fungal OTU. Now I am going to talk some about some indicator species analysis. This indicator species analysis represents some rare or some indicator species with a particular geographical origin. For example, our analysis revealed that the higher distribution of higher number of indicator OTU was observed in PPN origin and in for fungi and TV region for bacteria. And also we found that the strong basidiomycota OTU indicator of high esters and TNC condition were predominantly observed from the PPN origin. In contrast, the Ascomycota indicator OTU of low esters and TNC conditions associated with BK origin. Previously, in biochemical parameter, I already mentioned that this PPN origin shows the higher esters and TNC condition. This is a very interesting result because of some fungi may have symbiotic nature and some is more rather than the endophytic nature. So maybe in our study, most of the Basidiomycota OTU in the species level is symbiotic nature. So when plant produce or store more TNC or esters, then the symbiosis is reduced. So this result is quite interesting to me in my study. As in relation to the previous slide, here also we can have a look that the Basidiomycota OTU is negatively correlated with root esters and TNC because most of the Basidiomycota OTU is in species level, it is symbiotic in nature, but Ascomycota is positively correlated. And this pattern reflects the differential response of Basidiomycota and Ascomycota to plant-generated carbon metabolite, likely indicative of their symbiotic and saprotropic nature in different origin. In the bacterial abundance, the Acidobacter is negatively correlated with the root esters and TNC, but Bacteroideata is positively correlated with root carbon and nitrogen. Negative and positive correlation of bacterial phyla, Acidobacter and Bacteroideata, respectively, with root esters and TNC, indicate differential response of this bacterial group to plant-produced carbohydrates. Now I am moved to the beta diversity index. We have found the significant influence of tree origin and season in the composition of fungal and bacterial community. And there was significant correlation with this community with root biochemical parameters, for example, root esters, root carbon, and total non-structural carbohydrate. And here is also the mean L1 temperature and precipitation of the geographical origin is correlated with the fungal community composition. This may be because of the evolutionary adaptation of this geographical origin plant because of the diverse climate condition in different geographical origin. And also it is because of previously I mentioned that their relative abundance is affected, so it influenced the whole community composition. And I have also constructed the co-occurrence network. Maybe it's difficult to understand this figure, I know, because this is a very large figure and I put all figure in together. But here there is some is dot and some is square. So dot represents the bacterial OTU and square represents the fungal OTU. So I have generated the co-occurrence network with minimum threshold of R square value is 0.9. And here is the dot means OTU. And dot or square means OTU, and the line means the connection or significant correlation between fungal and bacterial OTU. So I will give you the detailed result in the next slide extracted from this network graph. So the co-occurrence network, I said the number of edges means connection. And this is the degree of co-occurrence. This data is extracted from the previous network graph. And this is the number of node. Number of node means significantly number of OTU correlated with fungal or bacterial OTU. So here we can look that in beer region exhibited the higher number of edges and higher number of node and the average degree of co-occurrence. This higher number of node in both season in beer region indicator of a broad spectrum of endophytic OTU that co-occurred or cooperation. And also the presence of higher number of positively associated bacterial and fungi in beer network point to increased cooperative and interaction event. I have also conducted some analysis how the fungal phyla and bacterial phyla is correlated, because previous slide was based on the OTU, but this is based on the phylum level. So this analysis was also done based on the co-occurrence network matrix from the previous analysis. And here the circle represents the co-occurrence frequency, and the chord thickness represents the co-occurrence strength between fungal and bacterial phyla. And we observed diverse interphyla co-occurrence pattern across different origin and season. For instance, the frequency and strength of co-occurrence between Ascomycota and Basidiomycota with firmicutes were notably higher in beer region during both seasons compared to the other geographical origin. This is because I already said that some region have originated from the higher precipitation or lower precipitation. So the trees originated from the higher precipitated area produce more photosynthetic carbon, for example, esters and total non-structural carbohydrate. So it also reduces the cooperation for nutrient exchange to the plants. So now I'm going to the conclusion. This study established that the composition of antibiotic fungi and bacteria in Pinus sylvestris tree is shaped by both tree origin and the season, and is closely linked to the root biochemical trait. The observed variability in relative abundance of key fungal and bacterial taxa across different seasons underscores the influence of tree origin on microbial community dynamic. And finally, the co-occurrence network of fungal and bacterial endophyte in Pinus sylvestris exhibited variation in network complexity across geographical origin and season. Finally, we can take the message to the home that thus the ecotypic difference in host-root biochemical phenotype driven by evolutionary adaptation to geographical and climate condition play a pivotal role in shaping root endophytic community assembly of Pinus sylvestris. I must show my gratitude to Professor Joanna Muha, who secured funding for this, do this analysis, and also Professor Katerina Rinkiewicz and Professor Agnieszka Shuba for their help in suggestion, contributed greatly to the work. My sincere thanks to go to everybody to the Institute of Dendrology for their support through the course of the research. Thank you for your attention.
Thank you very much for your presentation. Now we will skip to totally different area of expertise. And Dominika Robak from the Institute of Dendrology will share her insight into the topic of natural regeneration of black poplar along the Vistula River Valley in Poland.
Good afternoon. The topic of my presentation is natural regeneration potential of black poplar along the Vistula River Valley in Poland. At the beginning, I would like to say a few words about black poplar, which will allow you to better understand the topic. Black poplar occurs in almost all of Europe, in Central Asia, Northern Africa, and Western Siberia. In Poland, black poplar reaches its northern limit of range. Black poplar is a wide-pollinated dioecious species with male or female individuals. Red-hanging catkins that produce yellow pollen in large quantities are males. Green hanging catkins that develop into white fluffy fruit heads are females. Light seeds are dispersed by wind and water over long distances, resulting in high migration rate, high gene flow, and genetic diversity. Specific water and soil conditions are necessary for seed germination. This means that black poplar requires moist soil and a sandy area. Black poplar is a main component of riparian forests, extremely important both from ecological and economic point of view. The habitats where it occurs are considered centers of biodiversity. Black poplar is characterized by high plasticity and tolerance to pollution, so is often planted on post-industrial places. Because it adjoins to riverbanks, it's considered natural flood protection. Poplar, as a fast-growing tree, is an excellent source of biomass for energy purposes and also an intermediate product for furniture and paper industries. Over the last decades, we observed a drastic decline in the black poplar individuals in Poland and also around the world. Progressing urbanization process, river regulation, and transformation of floodplains have led to the reduction of extremely important areas of floodplains that provide river sediments which are necessary for seed germination. Therefore, the lack of suitable areas results in lack of natural regeneration. An additional threat is the fact that black poplar may cross with fast-growing hybrid poplar varieties, which may lead to the contamination of its gene pool. Our research covered a total of 623 black poplar trees. We studied 4 different populations located along 4 different sections of the Vistula River in Poland, each population comprising a group of mature trees and a group of natural regeneration. The aims of the research were to determine species purity and clonality, assess the genetic variability, compare the gene pools of populations, compare the gene pools of potential natural regeneration with the gene pool of mature trees growing in the same location. We also wanted to check whether the selected reproductive strategy differs depending on the location and degree of land transformation, as black poplar may also reproduce vegetatively. When it comes to the methods, the first step was to collect leaves from the tree. The next step was isolation of genetic material. Then we amplified species-specific nuclear and chloroplast DNA markers. Then we amplified nuclear microsatellite markers. The last step was genotyping on an automatic capillary sequencer and data analysis. We identified hybrids based on species-specific nuclear WIN3 marker. It allows us to identify hybrids of the first generation. In the case of pure species of black poplar, after the PCR reaction, we obtain one PCR product visible as one band on the gel. In the case of hybrids, after the PCR reaction, we obtain 2 products visible as 2 bands on the gel. Our results showed that in the group of mature trees from the middle section of the river, there were 9 hybrid individuals, and in the group of mature trees from the lower section of the river, there was 1 hybrid individual. We also identified hybrid based on species-specific chloroplast marker. It allowed us to identify hybrids of the second generation. And the PCR reaction product is digested with the restriction enzymes, thanks to which we obtain a specific pattern of bands, specific for hybrid individuals and specific for pure species. This analysis confirmed the previous results. The analyses were performed based on the polymorphism of 14 microsatellite markers. Amplification of these markers enables genotyping, which allow us to know which individuals are clones. Here we have a picture obtained from an automatic capillary sequencer, and we can see that the presented individuals are clones. Here we have a table presenting the parameters of genetic diversity. The parametric of genetic variation were generally comparable between groups and also comparable to the other different studies carried out on the populations in Poland and also in Europe. We observed high genetic variability. The genetic differentiation among the studied group of natural regeneration was almost twice as high as among the group of mature trees. The highest percentage of clones was observed in the population from the lower section of the Vistula, located closest to the river influx, where clones constitute 94% of the population. And the lowest percentage of clones we observe in the population from the middle section of the river, where clones constitute 3.5% of the population. Only one natural regeneration turned out to be a true generative natural regeneration. This is because only in the middle part of the Vistula there are conditions for seed germination, because this area is least transformed by human. Population from the lower and upper section of the river are in the worst situation because this area is heavily transformed, and natural regeneration turned out to be half vegetative, half generative, or fully vegetative, as in the case this group located closest to the river influx. A high number of private alleles in the group of natural regeneration from the middle section of the river, maybe because this is the only one group through generative regeneration. Here are the results of principal coordinate analysis and showed us that the population located closest to the influx of the river was the most different from the others. We can see it on the picture A. This is this orange point. At the individual trees level, picture B, C, and D, we observed that only in the middle section of the river, the group of natural regeneration reflects the gene pool of mature trees group. This is on picture C. For the remaining populations, on B we have population from the upper section of the river and the lower section of the river. So in the case, this both, the group of natural regeneration are less similar to the group of mature trees. Here we have the results of genetic clustering, which confirmed the previous analysis. We analyzed two approaches. The first indicates that the population from the upper section of the river constitute a separate genetic group. And the second approach indicates that the population from the upper section of the river and also from the lower section of the river constitute separate genetic groups. This result indicate that there are some limitations when it comes to the gene flow throughout the Vistula. To sum up, black poplar populations in Poland are usually old and occurs mainly in small groups. Moreover, many rivers are significantly transformed, resulting in a lack of suitable areas for natural generative regeneration. Generative regeneration occurs quite rarely, although natural regeneration still occurs in most places where the natural environmental conditions allow it. It is vegetative regeneration, the vegetative type of reproduction which enables the survival of local population in areas unfavorable for seed germination, reduces genetic variability, and thus the adaptive capacity of the species. This is a big problem considering the rapidly progressing climate change. The analyzed populations are characterized by high genetic diversity. Genetic differentiations among the analyzed groups of mature tree is high. We think that taking actions to ensure the protection of the black poplar in Poland seems to be necessary. And thank you for your attention. That's all from me.
We have now the last presentation of this session, because this session is a little bit shorter than the previous ones. And I invite Paulina Pacek from University of Environmental and Life Sciences in Wrocław. And Paulina, we talk about aliens among us. This alien is called Coletotrichum. Coletotrichum, yeah? Yes. So please start.
Okay, hello everyone. I'm Paulina and I'm a master's student, and I came here from Wrocław. And today I would like to speak a little bit about invasions. And this slide, obviously, I put it here to wake you up a little bit. I know we might be sleepy after yesterday's party or today's lunch. So this slide is just to catch your attention. But also I think for a regular person, when we use term invasions, maybe such pictures might come to their minds. But obviously for us, for people who are associated with science in some ways, when we think about invasions, we think about mostly a threat that it can pose to native, not only species, but also ecosystems. So there's term invasion, but there's also many other terms that are, maybe I'm not close enough. There are also other terms that are associated with the term invasion. And one of them is spillover. And these terms describe a situation when alien pathogen is transmitted by invasive species to a new area. And this can threaten native species. And please remember this slide. I will come back to it in just a second. And now for our Coletotrichum acericola, which, as was mentioned, it's the potential threat to maple trees, to native maple trees in Poland. So in 2023, this fungus was isolated from Acernegundo. Most precisely, it was isolated from the fruit of Acernegundo. And as we know, it's an invasive species in Poland, very invasive. And it was isolated from the fruit of Acernegundo, and the symptoms of the infection can be seen here on this picture. Those are just dark spots. And this species belongs to Coletotrichum agave species complex, which is quite interesting because if we compare the morphology of this species and also different species within this complex, we might find there are many differences between them. So that's quite interesting. And then we move forward to how this fungus develops, grow on a medium. So on PDA medium, as you can see here, we see orange mycelium in the center, and also we can observe white earlier mycelium with a white margin. And what's interesting is that here on the left side, there is a different type of medium used, which is maltose agar. And as you can see, this is exactly the same strain. But for this medium, it develops a different phenotype, which I found quite challenging throughout my whole research because, for example, after 3 or 4 passages, I've noticed a change of phenotype of the same strain, so I had to be really careful. Okay, so coming back to this slide and spillover. So Acer negundo is an invasive species of tree, and it came from North America to Europe, as we know, into Poland. And so the spillover was the main key idea of my research. I asked myself a question. So since Coledotrichum acericola was isolated from invasive species of Acer negundo in Poland, in Wrocław, by the way, so I asked myself a question, does it pose a threat to native species of maple tree, Acer pseudoplatanus and Acer platanoides? And in order to answer this question, I conducted pathogenicity assays. And here I presented how it looked to make it easier for you. Because, well, yeah, starting with the top part of this chart, as we can see, I used leaves. And what I did, I cut out a small part of mycelium of Coledotrichum acericola, and I put them on the leaves. These are the leaves of Acer pseudoplatanus, platanoides, and Acer negundo also I used. And then I waited for 2 weeks to see if any necrosis was developed, which means we could observe an infection caused by Coledotrichum acericola. And after 2 weeks, if I observed necrosis, I cut out the necrosis. I put them on PDA, medium potato dextrose agar, and then I waited around 2 days to see if Coledotrichum acericola was the reason why I noticed necrosis on the leaves. Similar situation with shoots, because I also used shoots in my research, but only Acer negundo shoots. And the difference between this part of experiment and this is just here, when it comes to shoots, I used conidial suspension rather than like small parts of mycelium. And then I again waited 2 weeks and cut out necrosis to confirm Koch's postulates, which is a gold standard for microbiology. So as you can see, this method it's not very difficult to conduct in a laboratory, but little did I know how much of a struggle I will face during my research. So as you can see here, I'm showing you infection, the leaves with visible infection after 2 weeks. And as you can see, everything looks fine. We might think the necrosis that you can see here on the leaves, the reason for it was Coledotrichum acericola. Well, it's not that easy. Why? Because this is what I got when I cut out the necrosis. As you can see, it doesn't look like Coledotrichum acericola from the previous slides that I showed you, which is usually orange mycelium with white aerial mycelium. So what went wrong? Well, one reason is quite actually simple. I don't know why I didn't think about it before starting my research, but well, when I started my research, I knew that I have to sterilize the leaves somehow to get rid of any other fungi that obviously lives on the leaves, and I could not get rid of them. So this is exactly what happened. You can see that I isolated some Alternaria alternata, which is obviously a fungi that can live on leaves. So yeah, this is the result that I observed the most. But also, I managed to isolate some strains of Coletotrichum acericola. Currently, they're being I will get sequences next week, so I would be able to tell you 100% that this is Coletotrichum acericola. But from microscopic observations, we said that these strains, Coletotrichum 2 and 1 and 4, was the reason why I noticed necrosis on the leaves. Next, coming to shoots. With shoots, the situation was quite easy because, well, as you can probably suspect, we don't have that many fungal community on the shoots comparing to the leaves. So that was definitely easier for me to conduct pathogenicity assays on the shoots. So here you can see the growth of fungus after 2 weeks of inoculation. And the growth is visible. And also you can see necrosis, which, as I said, I then cut out and put on PDA medium to confirm Koch's postulates. And here is the results of the assay conducted on shoots. You can see that those are just some examples, but I can tell you for now that the pathogenicity test on shoots was much more successful than on the leaves. So yeah, here you can say, for example, that the strain Coletotrichum acericola number 7, I was able to re-isolate from the shoots, so that's a success, as well as Coletotrichum strain number 9 and number 4. So what I would like you to remember from this presentation, well, first thing is that the problem with invasions, it's not only the invasive species that came from different parts of the world, for example, to Poland, but also there is a problem with pathogens that these invasive species can carry. And I feel like sometimes we forget about this part, so I'm glad that I was able to mention this today in my presentation. And if you ask me, so please answer the question: does this fungus Coletotrichum acericola pose a threat to maple tree species or no? I can tell you for now that based on this, what I got is that yes, but I cannot tell you to what extent this threat is, and I cannot tell you for now any predictions about it. Hopefully, I will continue this research, and maybe on next conference I will tell you more. And thank you very much for listening. That's it.
Yes. Thank you very much. And I would like, first of all, thank to all the presenters for sharing the research with us. It was a very interesting session for me personally. I would like to open the discussion now, so please free to ask any questions to our presenters in any order you would like. Yes, please.
How do you remove microorganisms from the leaves without destroying the cells of the leaves?
So you're asking me regarding the pathogenicity? Ah, okay. It's It's actually very simple. I just use hypochlorite sodium. It's you can find it any lab. You can also use ethanol. Oh, and actually, sorry. What's important, it's the time that you sterilize the leaves, because maybe I forgot to mention that I only sterilize the leaves for 30 seconds, and as you can see by the results, it's not enough. I was not able to get rid of the fungi that I was not interested in. So I'm gonna actually repeat the assays, and I will sterilize the leaves for maybe 3 minutes. I will try, and I will see if it gets any better.
I have a question for Kasia. If I understood correctly, you choose fruits as a fecundity indicator, yes? Yeah. So don't you think it's quite limited, since some fruits may be seedless and may not contain a living embryo? We observe in the Institute of Dendrology, in the case of European beech or Norway maple, that they may have a fruitful year, but in case of the seeds, it's not so easy. When we want to get the material for study, it turns out that there are no seeds. There are fruits, but there are no seeds.
So it actually happens really rarely. There aren't any seeds. What we do, we usually randomly select 5 inflorescences for each tree, and we count the seeds. And it usually runs from 2 to 5 seeds. And as you mentioned about the beech, because I'm also working on the beech, it's true that in the beech, seeds can be just virtually empty, even though you get the seed. And with drawing, we didn't really see something like that. So I wouldn't say it's limiting. I think that counting fruits is a way to go. So yeah.
Lucas, so out of all alien and invasive species in Lithuania, which one causes the most problems?
You mean in general or just of the genus Kórnik? I think, or about, you are asking about woody species or any species. So I think it's Heracleum sosnovsky, which is a very huge problem in Lithuania because it was planted during Soviet times as a fodder plant, and now we have areas where it covers thousands of hectares in the east part of the country, and it is estimated that the eradication of the species in such parts can cost several million euros. And I think, and it also causes ecological, economical, and social issues. I think this is the most invasive species in Lithuania. And from the woody species, I think Robinia pseudoacacia is also a huge problem, especially in the western part of the country, in Kórnik and Spit, where the whole site is UNESCO heritage site, and these habitats are very susceptible. And Robinia causes alter soil properties, and these habitats are changing.
So, Paulina, so this Colletotrichum fungus, how severe is its infection? Can it kill the tree, and does it cause a lot of problems in America?
Well, actually, as you could see on the slide, it causes dark spots on the fruits, and as I showed you, it could also infect the leaves, but we don't know yet to what extent. And in America, it seems like this fungi doesn't cause any severe symptoms, or it doesn't develop a severe infection. So no, as I know, the trees are not dying in America because of this. The Acernegundo doesn't, and so. But yeah, we will see in a few years how is it gonna go in Poland. Maybe the infection will be much more severe.
And any idea how could it be carried to Poland? Because we know Acernegundo from many years. And this fungus appeared right now. So, what it could be your idea about the transport?
Yeah, so probably it came with the seedlings. This is I think the most reliable hypothesis that I've read about regarding this species. Yeah, but also I read that it's possible that in North America it was always on Acer negunda, but it was just no one actually cared about it, and then someone actually my supervisor cared, and she managed to check if this fungus actually appears in Poland. So it's also possible that it was here on Acer negunda for many years, but we just didn't check it. Yes. So the reason how it came, it's still not. We still don't know yet.
Paulina, I have a question for you because I want to ask you that you found this pathogen only on Acer negunda, or this pathogen also jumped to our native trees, any others?
So yes, it was isolated from Acer negunda fruits in Wrocław, and that's how the species is described from Acer negunda. But yeah, so the aim of my research is to check if it poses any threat to our native species of maple trees, but for now, no, it was not. It was never isolated from our native species. So we may suspect that. Yes, for now.
Again, for Paulina, sorry. Do you know, are there any more cases from Europe of the species?
So, as far as I know, the fungus was only isolated in Poland, but back in my lab, I have fruit with visible symptoms from, I believe, Germany and some other parts of Europe, but close to Poland. And I still didn't have time to work on them. But the symptoms are visible, and also under the microscope, it looks like this is fungi, but it needs a little bit time to conduct this research. But yes, but we have such samples.
Yes, again, the question to Paulina, because your presentation was the most understandable for me. I have a question about the names of the strains that you isolated. Col 4 and Col 7 and Col 9, and Col 0 is widely used wild type of Arabidopsis thaliana. Aren't you afraid of a mess with such names?
Well, I've never thought about it. You just said it. Well, Colombia, yes, as I know. Well, I don't think I should be afraid, but I don't know what to say. But well, I will definitely think about it. Yeah, but I think it's big because these are different kingdoms, so I don't think it's gonna be it can cause any problems with someone will say, oh, this is the fungus, no, this is Arabidopsis. I don't think it's gonna be the case, so I think I'm safe for now, but I will keep that in mind. Thank you.
Okay, fine. We have also other presenters, I remind you. Okay, I have two questions for Lucas. One of the first slides, you showed us pictures of different invasive species in Lithuania. I think I recognize Sorbaria in one of them. Was it Sorbaria sorbifolia or Kirillovi?
That's a very good question, actually, because Yeah, we found a species that looked different from Sorbaria sorbifolia. It was much larger. It was taller. It had larger inflorescence. So we might think that it's Sorbaria kirillovi. Yeah, yeah. Thanks for confirmation.
It looks like it needs to be, examined. And my second question is, to what you can attribute the fact that this Cornus sericea and alba produce more flower and resulting in less fruit? Was it be like a lack of pollinators or something else?
I think it might be a bird factor because it seems that Cornus sericea produces different colored fruits and maybe they are more attractive to birds. And also Cornus sericea occurs more closely to natural habitats, and maybe the more birds are present in these habitats, and maybe they pick the fruits, and maybe this is the cause.
So it's actually not fruit production, but what you can find after some time on the. Okay, fine. So okay, fine. And may I have another question for another presenter? Yes, I have something. Oh, okay, so yeah, I have one question to Polak, and because it appeared one word that this is very new for me, formicutes. What is it?
Firmicutes is a bacterium phyla. Yeah, bacteria is bacteria, because we have bacteria and fungal phyla, so we make some co-occurrence. Yeah, thank you.
Any other questions before I ask? Okay.
Okay, thank you. I have a question to Kasia. Because you was talking about the neighbors between your trees, so I would like to ask you, so it's your own method to count this, and also if you count it, you choose only the trees too close to your tree or some distance around the tree?
So no, it's not my method. It was one introduced by a researcher called Contreras. And I should have mentioned that, but we've only taken into consideration the trees in 10-meter radius. So yeah, that was the distance.
Dominika, I have a question for you. Can you confirm that black poplar can be one of the most threatened tree in Europe because of habitat loss, hybridization, and climate change?
I don't know if one of the most, but in many European countries, black poplar is considered as an endangered species. In Poland, not yet. Is endangered probably in Great Britain, in Germany, but I think now there are many species which need help.
Any other questions? Okay, this is my turn. Okay, okay. Dominika, you mentioned the contamination of the gene pool of the black poplar. I was interested in two aspects of this. One of this, how difficult was the identification of the black poplar in the field? Because I assume that you didn't collect all the poplars all around. You selected somehow intuitively more or less pure species. Yeah.
To recognize hybrids in the field is very difficult. It's difficult to distinguish it based only on morphological or phenotype. So we always have to collect leaves from all the trees, and then in laboratory using this species-specific marker, we can assess that it is pure species or hybrid.
another question goes to this relation because if we talk about hybrids of black poplar, there are more than one species, American species of poplars in Europe, so it can hybridize with different of them. I mean, this is like a pool of everything now mixed in Europe. So how specific are the markers you use? So how can you be sure that this is really pure or hybrid species? What do you think?
I think they are very high specific because we have one chloroplast which is inherited from mother, and we can check the hybrids of the second generation. The next one, WIN3, which can show us the hybrid of the first generation, and also we can forget that we have microsatellite markers. I used 14, but it's more, which also can show us that the individual is pure species or a hybrid.
Because we know that hybridization, because poplars is very, very easy. And so do you have any data, maybe not from your research, but in general from the literature, how often it occurs? Because, everyone mentions that it's easy, but in numbers, do you have any numbers about it?
In the literature, it's not so often, and it seems to be more rare because the time of flowering, because of the progressive climate change, is different. So probably there is less risk when it comes to the hybridization.
It's common, but not that common that we could be, afraid of. Any other questions, please? If not, I invite you to the coffee break, probably not to lunch, not another lunch, as it is in our schedule. And please go and be first, first come, first served. So thank you very much.
I would like to introduce Niccolò Di Marzio, who will tell about extension of skid tray network on salvage lodging operation, the study from Italian Alps. The floor is yours.
Put it here.
Oh.
And should.
Okay.
Okay, so hello everyone. I'm very happy to be here to introduce my newest research project. My name is Niccolò Di Marzio. I'm a PhD student from University of Padova, and I work in the forestry and forest mechanization sector. I would like to start this presentation by quickly and briefly introduce the framework of my PhD project, as it's taking advantage of the switch of forestry sector from a traditional systems relying on manufacturing and logistic to something more specific with connected and integrated solutions that we call also Forestry 4.0. And in particular, the main focus of my PhD project is the management of high-resolution data, such as satellite image and laser scanning image and data, in order to both put into practice in order to plan forestry operation, and in particular, because as you know, Italy is a highly diversified country, so going from the sea level up to the alpine region, we can put into practice site-specific planning, and as the case that I'm going to present now, we can also use this high-resolution data to put into practice post-harvesting monitoring. In particular, today, I'm taking advantage of a very extreme event that happened a few years ago in Italy that you probably, I think you probably heard about it. That was the Vaja storm in 2018. There was a similar event in Poland, maybe even bigger, in 2017. So the Vaja storm was an extremely severe weather event that struck Italy during the night at the end of October, and it devastated mainly three regions that are Trentino Alto Adige, Veneto region, and Friuli Venezia Giulia, but also other regions around. It was characterized by strong winds and heavy rainfall, and this storm, as you can see from the picture, also destroyed many infrastructures, electric lines, and there were also people that died, and there were millions of trees that were damaged and uprooted. So the damage was also quantified in terms of billions of euro, calling for a highly extensive recovery and reforestation effort. And also this storm highlighted the vulnerability of the land and the need to enhance the resilience mechanisms for in view of the climate change. So just to give you some numbers, as you can see, this storm was during the night between 29 and 30 of October. The winds were in many parts also over 190 kilometers per hour. It destroyed something like 8.6 million of cubic meters and a surface of 41,000 hectares. You can see from the percentage of damage that in some parts, such as the Veneto region, which is the one I'm representing now, and Trentino Alto Adige, in some cases, the damage was around from 20 to 50% of the forest area. In this project, the main focus is to to make a characterization and examination of the intensity of salvage logging operations that were put into practice after the storm. And specifically, we focused on the ground-based extraction system and with high level of mechanization. So no animals, no agricultural tractors, no cable lines, but we just focused on the highly mechanized ground-based system, which is harvester and forwarders. As you know, the salvage logging is the removal of trees that is carried out with the main intention to recover as much as possible the economic loss, and also to reduce the hazards to the infrastructure, and in some cases also to ensure the human safety and the risk of following disturbs. Because I didn't show you in this previous pictures, but after the Vaya storm, another problem that came, which is of course connected to it, is the bark beetle attack, that now it has been quantified that made even more damage of the Vaya storm. So the numbers that you see here that are related only to the storm, you can think that it's now the damage is also twice those numbers. So on the one hand, salvage logging is the most widespread management response to this kind of disturbance worldwide. On the other hand, salvage logging can also bring some negative effects on the biodiversity, or it can affect some mechanisms that are related to the ecosystem resilience. So when it comes to talk about salvage logging, as you know, it's always a little bit controversial topic. So the analysis that we carried out focused on two main study areas. One is located in the south part of Trentino, and one is in the north part of Veneto. So we selected 12 forest sites in Trentino and 17 forest sites in the Veneto region. So in order to carry out this analysis, we went through this methodology, and then I'm presenting the preliminary results of this. Of course, then we need a second part of analysis that is ongoing right now. So we started with a digitalization of all the forest tracks that we could observe by satellite image and by orthophoto that were provided by the regional administration. We digitalized approximately, merging both study areas, 800 kilometers of roads. And this phase, of course, was the most time-consuming. It was very accurate, and we tried also to recognize the main roads and the secondary and the storage areas, which is this one that you see here. After this, we carried out the terrain analysis. So all the terrain characteristics through the digital elevation model that we can observe, such as the slope, the minimum and maximum elevation, the exposition, and the percentage of disturbed area as the ratio between the area of the forest tracks and the area of the forest site, the storage area, and then we went through the deeper analysis of tracks. So we analyzed the number of tracks, the average and maximum and minimum slope, the length of the tracks, and then we used a road slope function, which is simply a tool of QGIS, to divide, as you can see here, every single road in segments of 5 meters of length. And for each segment, we got the information about the slope, divided in the two components, one in the direction of the road and one in the transversal direction. And the slope and the elevation, and so on. And this will be the part of the statistical analysis. After this, I can show you another picture. This is an example of 3 different forest sites. They were the operations here were carried out in different moments and sometimes by different forest companies. This is this is some data. For example, we also analyzed the spacing, which is an important indicator that helps the space, the distance between 2 different roads. And it's in this case, we can see that it's around 10 meters, which is consistent with the kind of machines that were used. Then we analyzed, we created the depth-to-water maps. The depth-to-water maps that is a map that they were developed around 2007 in Canada first, with the aim to map the wetlands on a large scale. So after those maps were underwent a very long period of validation and intensive field studies. And now they are used to predict the land trafficability across different kinds of terrain. So those maps are now implemented to support the forest management and the forest planning. And they can help also, as in this case, to preserve the forest ecosystem, because they can guide the planning phase or as in this case, the post-harvesting evaluation. So we used 2 different scenarios according to different thresholds of water network extraction. The first threshold is 0.25 hectares, which is a scenario that is considered as wet when, for example, the soil is in a snow melting period, or when we have a prolonged rainfall, or if we just are in a wetland. And the other threshold to extract the water network is the 1 hectare, which is the drier scenario which is the red one, and it's related to winter or periods with the frosted soil, or if we are in summer or in dry periods that now, by the way, are also increasingly more frequent. So then we calculated the ratio between the depth-to-water areas and we calculated those ratio with the sites of every forest site. And then we also calculated the depth-to-water along the roads. And then we made a ratio with both the scenarios on according to the length and to the surface that has been considered 4.5 meters of the road, which is the size of a machine, less or more. After this, we calculated the extraction distance, which is the measurement between the place where the timber is harvested and the point where it's collected and transported. So it's typically roadside, and it's this orange area. Here we have a measurement. This is for the sites. The green one is the average value, and the red one is the maximum value, which is around 200 meters, which is not too high. It's okay. Is in the average. And then we carried out a trafficability analysis, considering 2 scenarios of slope between 0 and 30 percentage of slope and 30 to 60%. We used the cost function that allowed us to keep in consideration only the continuous roads, starting from the orange area. And the idea behind this is that we can consider that for slopes ranging from 0 to 30%, we can use machines, normal harvester and normal forwarders, without creating such a huge impact. And for the slope from 30 to 60, then we have to use some adaptations, such as the particular chains that you mount on the tires, or you can use other systems. So whenever we found those, whenever we found those areas, and we know that they use those machines, then we can make consideration about the impact. So, as a take-home message, we can see that the increase in disturbance is now demanding an increased attention to define the post-disturbance management action, and they have to take care of the ecological resilience. I would like to just quickly show the future development of this study, which is the calculation of a severity index that gives us the impact, the idea of the impact of the forest road and the forest harvesting, in this case, on the forest site. So we will use, for sure, the depth to water maps that we calculated, the terrain characteristic, the presence of dry or fresh material, the roughness of the soil, and the difference of them to calculate the depth of the ruts. And that's it. Thank you for the attention.
Thank you. Now I would like to invite Zofia Świeżewska, who will tell us about peatland restoration that correlates with sphagnum fuscum morphology, if I pronounce correct.
So, hello. My name is Zofia Świeżewska, and I study second year forestry on Warsaw University of Life Sciences. And this is my first ever public scientific presentation, so please don't be too hard on me. And during this presentation, I will present the results of a research titled Peatland Restoration Correlates with Sphagnum Fuscum Morphology, conducted while on an Erasmus exchange on University of Eastern Finland in Peat and Trees Research Group. So to start, peatlands, as defined by Cambridge Dictionary, are an area covered by dead and buried plants, of course also the alive ones on the top. And this is a picture from a pristine peatland that I visited when I was in Finland. And sphagnum mosses make up most of peatland biomass, and almost 1/3 of European peatlands are located in Finland, so that's why the research is mostly there. More than 50% of Finnish peatlands are afforested and drained, and only 13% of area covered by peatlands is protected in Finland. And drained peatlands, despite only covering 1.5% of Earth's surface, emit up to 4% of global anthropogenic carbon dioxide emission every year. When a peatland is drained, it loses its unique biodiversity, causing plants and animals specific for that habitat to become extinct or very rare. Sphagnum mosses from drained peatlands show lower abilities of storing water, resulting in decreasing the amount of water peatlands can hold in case of a natural disaster. As we know, they are becoming more frequent. So it's a much needed option. So here you can see a mined peatland that was first drained and then mined, and this is a pristine peatland. Because of having many branches, as you can see here, this is a peat moss, and dense capitulums, so the top part of the plant, consisting mostly of water-holding cells, sphagnum mosses can store up to 20 times their own weight in water. Sphagnum fuscum, the special species I was researching, is this brown one here. Grows on hummocks, like the raised islands. Here you have the wet peatland, and these are raised islands of drier, not dry, but drier parts of it. And because of that, it is regarded to react to restoration faster than sphagnum species dependent on large amounts of water. Now I would like to introduce you to the lab I worked in. because it was essential and crucial for my research. And first, because of the amazing and much needed properties of peatlands, Finnish Ministry of Agriculture and Forestry of Finland funds a lot of research on peatlands restoration through various programs. One of them is called Catch the Carbon, and this is the one that was funding the Peat and Trees group located in the University of Eastern Finland in Joensuu in central eastern Finland. And this is the group with which I worked for 2.5 months during my Erasmus exchange on a project called Recovery of Sphagnum. I worked with Professor Eva-Stina Tuitila, the head of the research group, and a doctoral researcher, Nina Kumpulainen, to assess the impact of restoration of peatlands on Sphagnum fuscum. The group led by Eva-Stina Tuitila works on and publishes about consequences of climate and land use change for peatlands regarding vegetation, microbial communities, carbon dynamics, and greenhouse fluxes, carbon dioxide and methane. You can see the effect of their work in publications such as the ones shown on the screen. They also research interactions between people and environment in different approaches. If you want to see opportunities of cooperation or more information about their work, you can visit their website, and you can also find it without writing the lengthy thing. So data and sphagnum samples were collected by Nina and her colleagues from 5 peatlands, on which 20 sites were located. The sites were then divided into drained, the ones that were drained for all uses. We didn't differentiate the uses. It could be afforestation or agriculture or anything else. Restored sites, and pristine, so natural and untouched by the human, mostly untouched by humans. And later, this is where I came to help with the research. We analyzed the data, collected more of it by calculating and measuring weighting, and everything you could possibly imagine you can do with such a small plant. And we got our results where we established measures of 6 different traits of Sphagnum fuscum. Here you have like the normal stand of it. And we measured stand density, frequency, cover, fascicle density, capitulum dry matter, and capitulum diameter of the sphagnum. Here, to save time, and because the 3 I will present are representative for all of the results I got, I will only present some of the results. So I will present you the frequency, the cover, the capitulum dry matter, and that's all. So now to move on to my results. The data was collected from sites located on 5 peatlands. Here in southern Finland, this is the whole Finland and Finland in Europe, if you need help finding it. It was collected from Stormosse, represented by a green dot, Lölleneva by a light blue, Kauhaneva dark blue, and the dots are also on the plots in all the result slides. Then Lähnalamissuo, the pink, and Torronsuo, the red one. The colors are not associated with these peatlands on a regular basis, it's just that I chose the colors. And this is where the university was located, where I researched it and analyzed the data. So on this graph, frequency of Sphagnum fuscum is showed on drained, restored, and pristine sites. Drained, restored, and pristine sites. It was calculated by dividing the number of plots with Sphagnum fuscum by the total number of plots, and multiplying by 100% to get the result in percents. Average shows that drained sites have much lower frequency than restored and pristine. And in both cases, this is a significant difference. I know it doesn't look at it like that, but I checked it like twice. Oh, sorry. Oh no. That's not this one. Oh yeah, it's this one. Okay, sorry. So I checked it using the ANOVA and 2K test because I wasn't sure, because you can see the difference, but it's not as significant as the tests show. So yeah. And what we can see here, even if for the whole cumulative data, it doesn't look so true. If we analyze only one peatland, like Stormosse in this case, you can see the trend of restoring the measurements to the pristine ones. However, frequency doesn't show us how much of sphagnum fuscum there was on the site, rather how often it appeared. So to check how much of it there was, I also checked the coverage on all of the sites. And it is presented as a percent of plots that sphagnum fuscum covered on a whole 10-meter plot. And sphagnum fuscum coverage decreased after drainage. It's very small numbers, but as it was percents, I couldn't decrease it and make it bigger. And this also, when you consider it for a single peatland, it's more visible than for the cumulative data. That could be because of the difference from south to north of the peatlands. And also the data that was collected, because it was collected during 2 summer holiday months, so it was a big difference. So you can see the trend of coming back to the pristine sites. That's very nice, and that's what we wanted to find. And then this measure is a representation of how much water can a moss hold in its capitulum. This part of it, if you don't remember. This is a very important measure for what I was researching, because that's what I wanted to find out. If we can restore the properties of the peatlands that store the water when there's excess rainfall and then give the water back when there's drought. And this measure is traditionally, and also here, described by milligrams to grams. It's because of the way you count it. And as can be seen, dry matter content of sphagnum fuscum capitulum is lower, so its water holding capacity is higher on restored and pristine sites than on drained ones. Also, these results were significant, and the difference between restored and pristine was non-significant. So that was the ideal result I could ever wish for. So I showed you 3 different measurements out of many. Most of them show a high resemblance of restored and pristine sites, giving hope that restoration of these 5 Finnish peatlands is successful, and that in future all indicators will point to having fully restored the previously drained peatlands, and that maybe we can use the same methods that were used there outside of Finland and on more peatlands. Another conclusion is that sphagnum fuscum is a good indicator of peatland well-being. There are many projects undergoing, researching new methods of restoring peatlands concerning carbon and methane emissions. However, we are lacking indicators to assess success of such experiments. My work could provide information necessary to establish a much-needed indicator. Together with researchers about other sphagnum species and vascular plants from various peatlands in Finland, Estonia and other countries conducted by the group I worked with, these results will help assess the already used restoration methods and develop new, better ones. I would like to thank Eva, Stina and Nina for guiding me through my first scientific research and welcoming me into their group and their country. And thank you for your attention. In the end, I want to say that going on an Erasmus exchange is not only working and doing research, but also gaining new friends, traveling, getting to know new cultures, and of course, since I was in Finland, seeing a lot of aurora borealis, as you can see in the middle. And I would like to express that Peat and Trees Group is open to cooperations and incoming students. So you can take a picture of their address if you want to visit them, or just write it somewhere. Thank you.
Thank you, Zofia. Now I would like to invite Marcin Mazurkiewicz, who will present the presentation about how to do phorophytes shape the diversity of dependent epiphytic flora in Central European lowland forests.
Okay, that's it. Yeah, sorry. Okay, so I'm Marcin Mazurkiewicz, and I'm here to present in name of my friend Beata Olesik. She unfortunately cannot be here because of some reasons, but we did this research with our supervisor Sylwia Wierzchowska, so I think I'm competent enough to tell you something about this topic, and it is a question: how do phorophytes shape the diversity of dependent epiphytic flora in Central European lowland forest. And first, I would like to show you our three main hypotheses. So we think that a tree type, which will be a phorophyte in this presentation, shapes bryophyte species richness. Also, we think that host tree traits such as thickness class influence the diversity and abundance of epiphytic bryophytes, and maybe physical and chemical properties like the pH or bark and water capacity shape gills of those bryophytes. So first things first, study area. If you were here yesterday, then you know this area. If you weren't, then I will describe it shortly. So we did this research in Białowieża Forest. It is, they were presented. Oh, thank you. It is here in northeastern Poland, at the border between Poland and Belarus. We worked on Polish side, and here is area of my study and study of Beata. And it is 144 squares in managed forest. We were working here only on oak hornbeam forests, and they are located in these red circles. There are these green, 3 shades of green on this map. So there we collected our data, and what did we collect? So we choose 4 species of tree which were growing there in Białowieża Forest, and it was Tilia cordata, Quercus robur, Acer platanoides, and Carpinus betulus. And we wanted to find, and we found 240 trees in total, and they were divided in 3 classes. The small or young trees were trees with 20 centimeters in diameter at the height of 130 centimeters. The second class were medium, from 21 to 40 centimeters, and the third class were the largest, from 41 centimeters without any limitation to the top. As I said, I will describe a host tree as a phorophyte and epiphytes as a bryophytes, because in our Polish realia, they're mostly mosses and liverworts. So what data did we collect? Each phorophyte we divided in 2 zones. Zone A, it was from 0 to 50 centimeters around the entire trunk circumference. And zone B, it was from 51 to 200 centimeters, also along the entire trunk. And we determinate proportion of epiphytes as a percentage of cover, which is a classical method in phytosocial sociology. Except data of bryophytes, we also collected bark samples for water capacity and bark samples for pH test. And here you can see our census sheet. This may be not so good visible, so I will maybe not describe it further. So there are mostly bryophyte species with cover and diameter of a tree. So coming to results, and hypothesis number 1, here you can see how the cover of bryophytes on the tree looks like. Here is 3 species, and it is divided into zone A, which is this lower part of tree, and zone B, which is this higher part. And as we can see, in every species, zone A have much more bigger cover of bryophytes. Here we can see also a plot with a species number, and we can see that there are no differences between those species. Maybe Tilia cordata carries a little less bryophyte species, but it's not so visible. And in total, we had not so much species. I mean, these species were very familiar, the same species we found on most of our trees. Yes. And further about species richness. Here we have some PCA ordination, and I know it looks like a mess, but don't worry, it's quite good. What we can see here is each dot is each tree with and each zone. Here it is not divided into zones, and we can see that according to species richness, all of our four phorophytes are quite similar. Maybe Acer platanoides is a little outside of the rest, but it is not so much. And what we can see on the right side here is and then the S, and what we can see here is a bigger mess. But this big red circle represents a group of bryophyte species which lives on this lower part of trees, and this circle inside this red circle are the bryophytes that lives in this upper part. And we can see that mostly in the upper part of trees they live the same species at this lower part, which is not surprising. Of course, we have some species that are more epiphytic, let's say, than the rest of them, and they are those trees which has more this 100% epiphytic, let's say, species are a little outside of this group. They are somewhere there. And once more about species richness, we tried to do some analysis, but within only these areas, these zones of tree. So here is from the upper part. We can see that, again, trees are very similar to each other, and again, maybe Acer platanoides is a little different than others, but it's not so much. And here we can see species richness again, but only in zone B, and we can see that in Acer platanoides it is much higher than, for example, Tilia cordata. So when we analyzed only zone B, then we found that maybe Acer platanoides has more species. So coming to hypothesis number 2, here we have 4 species and pH and how it differs between them. And we can see that, again, Acer platanoides is different from the rest. It has much higher pH than the other ones. Of course, there was also some differences between those three species, but as we can see on the box plots, it's maybe quite visible between Carpinus and the rest. But Tilia and Quercus are not so different. And here we have a plot which shows us how this pH differs according to diameter of tree, which we used as a proxy for the age. And we can see that for Acer platanoides and for Carpinus betulus, pH growing with age. And according to Quercus robust and Tilia cordata, pH is lower in this, let's say, older trees than in this younger, which is very important for epiphytic bryophytes because pH shapes the biodiversity. However, we did not find it yet on our study. And here is some results from water capacity. And about this water capacity between three species, we did not find maybe some significant differences. That's why there is no box plot. But according to age of our trees, again, we found that Acer platanoides, of course, again, growing according to water capacity, and Tilia cordata also, but Carpinus betulus and Quercus robur, the water capacity of bark are lower in older trees. And it was water capacity measured after 96-hour sinking in water. And there are maybe a couple of methods to measure it. We will try. I know that Beata did 2 methods, so maybe using this other method, we will have another results. We don't know yet. But here, how it looks like. Yeah, and at the end, why is this research important? As I said earlier, we wanted to know which abiotic and biotic factors shapes diversity patterns in forests. We wanted to assist in decision-making process in forest management and to better manage our forests in Poland. We think that dynamic planning on conservation measures and management of forest resources, and as it is then, we have almost 30% of our country covered in forests. And of course, it is important data for decision makers and forest management, the Ministry of Environment, the General Director of Environmental Protection, so on, and also for national parks to better manage their resources, because forests are very important, especially for bryophytes, biodiversity and bryophytes, biodiversity, sorry. And at the end, some acknowledgements. And bark measurements was performed here in Kórnik, in Institute of Dendrology. So thank you very much for that. Statistical analysis were performance in Białowieża School of Statistics. So thank you again. And this research was founded by research project, Młode Umysły, Young Minds project, from Wrocław University of Environmental and Life Sciences. And it is also a part of master thesis of Beata. So thank you to her university for this funding. And thank you very much for your attention. And I hope you won't have a question for me, because I'm not Beata, but I did my best. Thank you.
Thank
So now I would like to invite Maria Bukovala, who will present novel pheromone traps for monitoring Thaumatopea pitycampa.
Good afternoon. I'm Maria Bukovala, and I am a postdoctoral researcher in entomology in Agricultural University of Athens. My scientific interests are based on store product entomology, and the last 9 years I had the chance to work in several scientific projects, and this gave me the opportunity to expand my scientific interest in forest entomology. So one of these projects was the European Research Project LIFE, under the acronym LIFE PISA, which was carried out in Greece, in Italy, in Spain against the Thaumatopea pityocampa, and in Scotland against the Dendrology spini. The aim of this project was to evaluate novel pheromone traps against both species, and also we tried to control and apply the mating disruption method against adult of both species. And another part of this scientific project was to control the larval stages through trunk traps. I am going to present this pest. I am sure that people from the Mediterranean basin, from Greece, Italy, Spain, are very familiar with this pest. The processionary pine moth Thaumatopea pityocampa is a very serious pest of the family Pinaceae, which occurs in forest ecosystems, in urban and also in suburban areas. This pest can cause significant defoliation to these trees, to the pine trees, and it can also cause the death of the trees, especially when secondary colonizers such as wood-boring insects or fungi can occur at the same time. So this will spread the death of the tree. It can also, maybe in cases where the trees can survive from Thaumatopea pityocampa infestation, the growth of the tree is seriously affected. Another important side effect of this pest is the fact that the larvae of this species, as you can see in the first picture at the right, the larvae are covered with urticating hairs that has toxin. And this is the defection of this species. So when they feel that they are in danger, they release these hairs, and when these hairs come in contact with our skin or with pets, they create irritation. So as you can see in the last picture, this is me after a trap inspection of larvae. So I suffered a lot during that period. And of course, most people suffered a lot, especially in schools. In Greece, schools are full of pine trees. So children are full of these signs of irritation. And also pets, especially dogs, that are very curious, and when they see the procession of this pest, they try with their tongue to understand what is this, and finally they suffer from irritations. So here is the life cycle of this pest. This pest has one generation per year, and during winter, the larvae creates the nests in the pine trees, and at February and at March, it depends on the weather conditions, the early instar larvae leave the tree and go down to the soil in order to pupate, to jump to the other life stage of the species. So, Thaumetopea derives from the Greek word thauma, that means miracle, because during February and March, we see 100 of larvae of this pest creating the processions, and suddenly they disappear. So that is a miracle, because during summer, adults emerge from the soil, and we see again this pest. And after the emergence of adults, mating occurred, oviposition, and finally the new larvae emerged, and this cycle continues. Here are some in the circles. You can see the pupate sites of this pest. They pupate inside the soil. Concerning the control of this species, there are some insecticides based on the bacteria Bacillus thuringiensis, and also some other insecticides which are called insect growth regulators that targets the early instars, the immature life stages of the pest. But as you can, but it is not. In a forest, it is not so easy to be applied because these formulations will kill the target species, Thaumetopea pithiocampa, but will affect the non-target organisms. So, in this context, we should be based on the development of a reliable monitoring technique, but to monitor the larvae of this species, as you can see, is very dangerous. Here, it's me again. During the inspection of this trunk trap, I tried to protect myself from the hairs of this species, and I was like this. These pictures are before coronavirus. So we propose that the monitoring of adults is one of the good solutions in order to protect the pine trees from this pest. So the aim of the project, one of the aims of the project was to evaluate the influence of different trap types against the adults of Thaumetopea pithiocampa. And for this reason, we developed 4 prototype traps, and we compared them with the commercial prototype. The experiment carried out between August and October in 2016. The experiment carried out in Attica. I'm going to present the results from Greece, from our university. The experiments carried out in Ktima Sigrou, which is located in municipality of Amarousion. Ktima Sigrou, or Sigrou estate, extends over an area of 950 acres. Ktima Sigrou is one of the last natural forests in Attica, and its flora mainly includes the Halepian pine, which covers most of the forest, but also there are several other types of trees, such as cupresses, oaks, locust trees, eucalyptus, as well as some other shrubs and other plants or herbs. At the same time, in this forest, this forest is a home of a wide variety of forest fauna. So the first one, G-trap, is a commercial trap, and we developed the other 4 traps, fly sun. We designed the difference between fly sun trap 1 and 2 is the open of the hole. The second fly sun has the biggest open of the entrance, of the hole of the entrance. We created 4 normal pentagons in order to install the traps, and the distance between the traps was in the same block was 80 meters, and the distance between each block was 100 meters, at least 100 meters. The capture male, we baited these traps with the female pheromone, an artificial pheromone, in order to attract the males of this species. And twice per week we recorded the number of the captured males, and we removed from the traps. Another fact that we took into consideration was the position of the traps. In every inspection of the traps, we changed the traps clockwise in order to avoid the influence of the location of each trap. So we change in every inspection the position of the traps. So here are our team, Professor Cavalleratos, me, and some students that helped us during the installation of the traps in the summer of 2016. And here are some other pictures during the installation of the traps. And here some other pictures of the traps after the captures. And here are the results. This table presents the mean number of the captured males during in each trap check date. As you can see, the modified G-trap, the prototype trap, the captures were higher in comparison to the commercial and the other 3 prototype traps. Especially in September 5th, the highest number of the captured males was recorded in the prototype G-trap modified, where it was 98.5 adults per trap, while in G-trap the number was lower, as well as at the other traps. And in this date, as you can see, in one trap, 154 males were captured, where the total number of the captured male at that date was almost 400 males. And here are the results. These charts represents again the total number of the captured males during the flight period of this pest. Again, as you can see, G-trap, the modified G-trap was the best, followed by G-trap, the commercial. To conclude with, the most effective trap was the G-trap modified. As you can see in this chart, again, the mean number of the total of the captured males throughout the experimental period was significantly higher than the other trap devices, so we suggest this trap for further to be included in a large-scale mass trapping of this pest. After this project and after we obtained the knowledge on how to control a lepidopteran species in a forest environment. Now we work in another scientific, European scientific life project under the acronym EGMER. Now we are in the last year of this project. The participants are from Greece, Slovenia, and from Spain. The aim is to control the larvae of this species, of Limandria dispar, which is another serious defoliator in forests, in urban and suburban areas. Here are some trunk traps compared with the commercial. Here are some pheromone traps, and if you are willing to see these traps, after the end of this session, we're going to show you, if you are interested, of course, all these traps. And also, we have applied the mating disruption method, which is a promising method for lepidopteran species, especially in the forest. And this is, I would like also to inform you that our team, we are special guest, we are guest editors in the special issue of the international journal Insects. This journal has an impact factor of 3. It is fast in review process. It has a high standard journal and high quality and high visibility. So far, we have 11 papers have been published, while other papers are under consideration, and we welcome contributions dealing with agricultural and forest ecosystems about pest management. Thank you for your attention.
Thank you, Maria. Now I would like to invite Valerio Proceda, who will tell us about the new approach for selecting the most prudent forestry ectomycorrhization.
And this. Hi everybody, I am Valerio Proceda. I am a student of the second year of the master degree of forest management and agriculture management in digital agriculture management and mountain areas. I'm here today because I want to explain way to approach to the digital management of microforestry, in particular in the truffle cultivation. But at first I want to do, before to start, a short introduction. In the digital soil mapping, we can use 3 methods. A discrimination method, it's a method that can define the homogeneous, subdivide the map into homogeneous maps units. The continuous method, it's useful if you want to create a gradient map. The map it's following a gradient. In this example, the gradient is rainfall, winter rainfall, and the clusterizing method it's a method that we it's similar, something like the previous method that I talked before. In fact, it has the same pro and contro. In the first method, the discrimination method, we have the low spatial variability within the same class, because of sure you don't have a gradient, and in the continuous method it depends of your quality of the data you have collected and the model that you used, because it's affected. They are affected the process to elaborate the map. In this study, I used all of these three methods to create a synthesized map to establish what is the most suitability site to growing truffle, in this case tuber melanosporum. It's also called preciously black truffle in common name. So this is the necessary steps for creating a thematic prediction map. You can use the digital elevation model, DEM, the other types of maps, digital maps like pH maps or temperature maps in for the area of study, and you synthesize this informative information, this layer of information to create a synthesized map. For example, I used a Kriging interpolation. It's like a statistic method that you can predicted the value of a punctual data information. In this case, the punctual data information is characterized by the purple point, okay? And you spread with this method, you spatialized the information through the space of your area of study. Through an inverse pairwise autocorrelation of this of the distance of this punctual information into a variogram, semi-variogram. This is a flowchart of geostatistical analysis and the interpolation Kriging method for do correct Kriging interpolation. In fact, you must examine the distribution of your data, because if you have in this case a Gaussian distribution, you maybe use a Gaussian model to represent this data, you know? So, and then you also can remove the outliers, the trends, because they affected the model negatively. Okay. This is a semi-variogram, and in the left, the image in left side. In the right side, we have two map. One map is the prediction map of the Zinco, in this case. The other map is an error predicted map, because every map product with this method have an error that you can predict. with different methods like cross-validation and leave one out, then one out, and so on. Okay. The next step, when you have all of your maps, thematic maps, principal thematic maps, it's to do a ranking matrix. The ranking matrix is the matrix that help the technician to evaluate which is the hierarchy of the criteria. But this matrix, it depends of it's subjectively, because it depends of personal opinions. Because if I think slope, it's more important for the edaphic growing of a truffle. Maybe I assign a value, more important than temperature or rainfall or pH. So it depends. There is not a rule for this. It depends of the technician that you ask a questionnaire of this topic, of the cultivation of, in this case, tuber melanosporum, okay? And then you assign with this matrix, ranking matrix, a weight of the importance of the criteria, okay? Then with the QGIS tool raster reclass, I reclassified the Kriging interpolation map into discrete map. So divided in 3 classes. Each class is a value. It consists in a value. Okay. So when I do this for all of my thematic principal thematic maps, I use the raster calculator to do a weighted multiplied matrices. In this step, I multiplied the maps like a matrices, okay? And this is and then I reclassified the map. The map that I product with this raster calculator, it's in the background. You can see? See. Okay. Then I reclassified with the FAO suitability classes, with 3 FAO suitability classes. And this is the output. In this output matrix, we can see 3 different colors: the ciano, the violet, and the yellow. Each color represents one value, a value of suitability class for the edaphic growing of tuber melanosporum. Okay, so this weighted multi-criteria analysis GIS approach allows the technician to select target zones within the area of interest that are most suitable to the photographic optimum of the mycorrhizal inoculum plantlet, because here you choose to put the inoculum where it's in the pixel where is cyano or in the pixel where is violet. Of course, in the cyano, because in the cyano you have the edaphic characteristics that concern the growing of the inoculum of tuber melanosporum, and not in violet. Maybe in violet you can do a thinning. Maybe because when you can do a thinning, you modified the microclimate. And maybe you can do a more lightness, more temperature that the tuber melanosporum needs. Okay, the litters, remove litters. So this map help the technician to do the interventions, but in the oculated method, in the precision method. Okay. Thank you for your attention and have a nice hunt.
Thank you, Valerio. Are there any questions, comments on this topic? Can you stay?
Sì, bevo un attimo.
Yeah.
Chris, thank you for the presentation. Maybe you can go back to the slides. We have this opportunity, but because I didn't get the difference between the bad, good, and high suitability. Can you explain maybe once again if you explained it already?
Yes.
For the almost the yes, good, bad, and high sustainability. Can you?
The high suitability is derived by a process to reclassification of the multiplied matrices, weighted multiplied matrices. When you multiply these matrices, you assign to each matrix, raster matrix. You know what is it, a raster? Okay, you know what is it, a matrix? Sì. Eh?
Once more. Can you repeat once more?
Do you know what is it, a matrix?
Yeah.
Okay, so when you multiply the matrix, you assign a weight, a weighted coefficient of this matrix. Okay, and the weighted coefficient is it depends of the ranking matrix decision analysis of this hierarchy matrix. Okay. So when you reclassified this matrix, this multiplied and reclassified matrix, you can choose the value of your maps, output maps. Okay. In this case, the high suitability level of the map correspond at a number. You can choose the number of your matrix.
Like a threshold.
Eh?
The threshold, the range.
Sì. Yes. Tac.
So we have bad, good, and the highest is high, classified as high. Yes.
Yes.
Okay, maybe I
And
was misguided by the that you pointed is it good, bad, and at last high. So I think I got it now.
But you can choose. I selected 3 of these, but you can choose 6. Maybe. I don't know. I choose 3 because it's more easy to comprehend, I think, but maybe I'm wrong. Okay.
You are totally correct. I was only misguided by the
also the
lining up it.
Also the multiplication matrix I used for this example. This is an example. I used only 3 map. Only 3 coefficient of the raster matrix.
Thank you. Thank you for going back to the explanation. I think I got it. Thank you.
Okay. You're welcome.
Are there any questions, comments? No? So, thank you. So, are there any questions to Maria about novel pheromone traps? Yeah?
I don't know how familiar are you with the, this caterpillar, but maybe you know, I'm just curious, what is the reason they shape this funny procession, the chain? Do you know the reason?
They create this procession in order to find the ideal place to pupate. So the first larva is a female who is the leader of the team. So all these larvae are in one nest, and the leader leads all the team in these places. If you remember the picture from the soil, so if you see, if you remove the first larva, they cannot find the correct place to pupate. Yeah.
So only the first one is qualified enough?
Yes.
That's fine. Thank you.
This reaction on the skin looks really scary. And maybe you have some protection in this school or the public place from this caterpillar? Don't anything? Maybe they think about to change the pine to the other trees near the public places?
Unfortunately, they cannot remove these trees from schools or from other places of public importance. And unfortunately, teachers do not say to pupils not stay below under these trees during the spring. And also municipalities who are responsible, at least in Greece, one solution is to remove the nests from the trees. Because they are visible from December, and they prefer to keep them in the trees, and the people and all people that are walk around this area suffer a lot from this pest.
Other comments, questions? I have a question about how many traps was removed by the person walking there in the region, because I think it was open space when you put the traps. So people takes the traps. They are interested, curious what's inside, or because they know.
Yes, I know. They are very. People are very curious, especially when they see something new. Fortunately, we have hidden this kind of traps, but in another experiment that was carried out at the same time, the mating disruption method, we had used a delta trap. The color of these traps was white, and people were very curious. And one time I had found inside the trap some cookies. But fortunately, they didn't remove the traps. But in this project, in Izimer, which is carried out in Peloponnese, we have lost some traps because people believe, oh, this is a trap, okay, maybe I can use this in another pest. So we have lost in this project some traps.
Because it just came to my mind that you may print a QR code on the trap so people who are too curious can scan it and read it about it maybe. It's just the idea.
Yes, this is a good idea. Maybe we are going to apply in the next project.
In Poland, we put the information there, be careful, there are ticks. So, it's working.
When we use
with different
warning
message,
yes,
let's say.
People usually attract from this message and steal the traps.
Thank you. Are there any other further questions for this topic? No? So, let's go to Marcin. Are there any comments about diversity? We can ask him, he will try the best for sure.
So Martin Orbatka, can you tell us how many times you need to collect data from one tree or one square? Because it looks like a lot of data and a lot of work, so can you explain us?
How many days or how many?
How many times? How many trees you can make in one day?
Okay. Oh, so to be honest, the most time-consuming part of our research was to find this species of tree with exact diameter, and we had well, the first couple days were very easy because we just picked trees which we like, and then at the end we have gaps, for example, in very old oaks or in very young tilia. So this was the most time-consuming. Collecting data on bryophytes and collecting bark was quite fast. I think half an hour and all three was done. I mean, this lower and this upper part. And we were very quick, and there was 3 of us, so we are still very good team.
Have you checked chemical composition of bark with the age, how it changed?
Well, we want it, and we think that it might be something, for example, nitrogen, potassium, or something like that. But unfortunately, we have funding only for pH and water capacity, and this was a part of this project. So maybe in the future we will try to do also some chemical analysis of bark, because there must be something that shapes these bryophytes biodiversity, and we are not yet sure what is this.
Because Białowieża, it's really, let's say, outstanding territory. So I think the nitrogen or potassium can be the reason. Yeah. Do you have any comments? No? So now let's ask Zofia questions with...
I have a quick question, because you were working on Sphagnum fuscum, and I'm thinking, can we use maybe some other Sphagnum species as this proxy of restoration of peatlands? Because Sphagnum fuscum in Poland is quite rare moss. I know in Finland it's, as we saw, 100% of Sphagnum fuscum at the peatland is, I don't know, some dream of in Poland. So do you think maybe some other Sphagnum species we can use?
So they collected data for, I believe, more than 10 Sphagnum species, but I was processing all the data, and the only whole species I managed to do in this short time was Sphagnum fuscum. So that's why I did it. That's why I presented results for it. But we still don't have the results for other species, because they're collecting also data from more peatlands around Finland and Estonia. But I believe we can, but Sphagnum fuscum is the most appropriate one because it grows in the drier conditions, so it's faster to come back. So.
Okay. I have a question. Have you checked any morphological traits between those different conditions?
So the measures, oh yeah, of course, I presented only the measures like other, but I also checked morphological traits. I counted the number of fascicles and branches on a 4 centimeter part of the stem, and also the total length of the stem. We also checked like the stand density. It was done by putting a square over the stand and then counting how many specimens are inside. So we checked it, but I decided these ones had the most. I just took these ones because all the results were similar, but I decided these ones were the easiest ones to present. Maybe, well, I.
You didn't found any difference in morphology?
It was the same as you did.
Okay.
Like there were differences.
Okay, but similar trends.
Yeah,
okay. Okay. And. Oh, I'm sorry.
First of all, congratulations on a very excellent debut, Zofia. And I would like to ask, have you seen our, can you tell us how does it look like, the restoration of a peatland in Finland?
So they have a lot of methods. Unfortunately, we don't know which sites were restored by which method. At least I didn't get the data. I asked about it, but they told me they don't know yet. But most of the peatlands in Finland are restored because most of them were afforested. So they were drained and then there were trees planted on them. So most of them are restored by felling the trees, removing all the wood, and then like pouring water over it again, so covering it in water and waiting till this sphagnum regrows.
Other questions? No? Okay, so now let's ask Nicole. Skid-tray, lodging, savage lodging. Do you have any comments, questions? I have a question. Do you want to test also the soil compaction effect on the biodiversity of soil or plants?
Yes, during the data collection, we also collected data concerning the soil compaction. So we want to calculate also the soil bearing capacity. And by the difference, for example, of the digital elevation model, we can calculate how is the difference before and after the forest logging. And the salvage logging also now related to the problem of bark beetle. And then, yes, we would like to correlate those information with other data that we collected, which is the number of cycle of the forwarder. A cycle is when the machine starts and collects all the wood across the site and then goes back. So we have exactly the number of cycles and the amount of wood, so we can calculate the weight and create a model for the soil compaction.
Did you remove that wood, all of the wood, or something stays there to decay?
Do you mean during no, they mostly remove the dead wood, and then they cut also the dead plants that were killed by the bark beetles. They left, of course, the let's say the rubbish material, because they can't do anything with that. Maybe it's better to leave in the soil.
Are there any information about diversity of microbiomes in the soil after such kind of treatment, let's say deforestation and harvester working there? Because maybe because of the soil compaction, you will have that different biodiversity, different species grown on the soil. Have you know? Do you know?
You mean in terms of organisms,
yeah, in the terms
or plants?
In plants.
Yes, there is both the effects of compaction, but there's also an effect of mixing the soil. So this is also why I was telling that sometimes salvage logging is a bit controversial, because on a side it can compact the soil and create all the related problems of erosion, of loss of nutrients and so on. But on the other side, it can also mix the profile of the soil and sometimes even enhance the renovation.
You didn't check.
No, we didn't check.
So do you have any other comments to maybe somebody else? No? If not...
Sorry, I had a question.
Okay.
But it was spoiler from... but I have been showed in Poland. It was in the eastern part of Poland, an attempt of, I don't know, it's a restoration or a creation by zero of a peatland. I don't know if you have any information about this procedure, that they were just felling the trees. At that moment, they were struggling with the birch, because, you know, they have a high capacity to resprout. And then I wanted to ask if you have also worked in Poland in it was the eastern part.
So I haven't worked in Poland on that. I hope to do it for my engineer thesis, but I haven't yet started it. So I first need to ask people to help me with it. So I would like to research about it, and I know they use mostly the same methods as in Finland, so like felling the trees and re-wetting, but there could be some differences because of the climate difference. So, yeah.
So thank you all of the speakers, and thank you for staying till the end of the conference. And now let's have a break and see you on barbecue. Thank you.
Before you go for your well-deserved break, I have one small announcement regarding tomorrow, because tomorrow at 7. Kórnik Lake. We will leave all the luggage here. the workshop will start at the institute. After the workshop, the bus will bring you here. There will be dinner. You can take your stuff, and then whoever is able to go will go with us to the National Park Góry Stołowe. So that's all. Enjoy your evening.